Racura Oncology, our first symposium. Whether it's the first of many, I guess we'll wait and see how it goes today. But welcome to all of you. Thank you very much for coming along and being interested in the Racura story. Just a couple of housekeeping points. Toilets are downstairs and then just a big loop around to the left and back underneath. It's pretty obvious when you get there. We won't be stopping for any toilet breaks or anything like that, so just feel free to get up and go. We will be having Q&A sessions, so maybe that would be a good time to go to the loo if you need to. So just by word of a little bit of an introduction, we've been asked, well, why the symposium now? Why the name change, which some of us aren't very good at adopting? And the answer to that is, as I hope you'll see today, is that we've come a long way in terms of, well, really across many domains within the company. We've got new intellectual property. We now understand the mechanism of action. That mechanism of action is informing where we look, where we think about doing clinical studies. So really a lot has changed, and companies have histories, and we really want to encourage people to have a look at Racura with fresh eyes, not with any thought of how they may have perceived the company in the past. And we feel that we've got a lot to build on here, as we'll come on to. The mechanism of action really brings together all of the things that we're doing within the company, and I think it really is starting to make a lot of sense. So we're going to be hearing a lot about that. We will be making forward-looking statements, and as usual, somebody says something witty about it, I'm not going to bother. And we've got this program. We will be having Q&A sessions after nearly all the talks, not after Daniel's opening comments, which are coming up next, but we will try and fit in 5 minutes of Q&A and then a larger Q&A session at the end. If we need to steal back some time, then we might lose a few questions, but the team will be available afterwards to answer any questions, so please feel free to make use of them. They've been trained by ex-KGB operatives to avoid any difficult questions, but you can try. So, okay, well with that, it is my great pleasure to introduce Daniel Tillett, CEO and Managing Director, and a very large personality behind Racura Oncology. So over to you, Daniel.
Thanks, Pete. Welcome, everyone. Thank you for coming. This really is the launch, the official launch of the new Racura. So if you've known about the old race, forget everything and start again with what you know. So I thought I'd take a different approach. I don't want to go into the history of the company. It's not really relevant anymore, hence the name change. But it does make a really interesting point, which is the history has really created a situation where Racura Oncology is kind of a unique company, at least on the ASX and potentially internationally. And it's for a number of reasons that are really important from an investment and clinical perspective. The first one, which I think is super important, is that we're clinically de-risked. We know our drug works. We've known our drug has worked for almost 40 years. What we didn't know is how it worked. So it's been all the way through to approval for AML in France, worked in a number of different other tumours types as well, and that really gives you an opportunity to really look to the future with a lot of confidence. One of the things is we're not repurposing. The closest I can guess is called reviving. We're bringing this drug back for exactly the same indication it was originally approved for. In fact, at the same concentration, same dosing level, same everything, that it was the upcoming phase 3 AML trial will be basically identical to the trial that was run back in the 80s to get it approved. Most importantly, the clinical problems that needed to be solved, which there were many, were all those that are tractable. So one of the things that is most terrible about the pharmaceutical industry or the biotech industry is the fact that most drugs just don't work. Your chance of getting a drug all the way through from preclinical all the way through to approval is maybe 0.1%. It takes billions of dollars and is something that is, very, very fraught with risk. And the thing we have is the problems that we had to solve were all this kind of the simple, easy problems that needed to be solved. Not all of them were easy, but they've been easy to, they have been all solved. The other thing that we're going after, we're not targeting orphan indications typically, although we do have an orphan indication in the form of AML, but even that in itself is actually a massive indication because it's our pathway to MIC, and you'll hear an awful lot about MIC coming up in the rest of the talks. So we're going after very, very large markets, multiple tens of billions of dollar type markets. That's the sort of scale that we're going after. So the opportunity there is as big as it possibly comes. We have a whole series of feasible pathways to commercial return, and Pete will talk about some of those in his section. And most importantly, none of this would matter if we didn't have the team. So one of the things I'm, I guess, most proud of at Racura is the team that's been built. The people that are there are world-class, fantastic, committed, dedicated, and you'll hear from some of them today. Unfortunately, you won't get to hear from all of them, but you know, without the right people, none of the promise goes anywhere. So what I wanted to give you is just a brief sort of overview of how difficult it is to bring a drug to market. It is so difficult that I wonder why anyone tries. The probability is tiny, the cost is astronomical, and it takes decades to get there. And if it wasn't for the prize at the end of it, no, I don't think anyone would bother. And I wonder why investors bother. And the reason why is there are hundreds of different gates that any new drug has to pass through, all the way from the original science, the target, identifying the leads, all the way through to running all the various clinical trials that are involved in that process. And as a consequence of that, you know, it is an area that many investors avoid, and they avoid because of the risk that's involved. The challenge that we faced at Racura was somewhat different. We kind of had a hodgepodge of the hard parts all being done for us. All the clinical studies have been completed and obviously made it all the way through to trial. We actually had a fantastic drug, so we didn't have to worry about the drug, but we had a real problem in that we didn't actually understand how the drug worked. People just assumed it worked in a particular way with absolutely zero evidence to support it. And we had a whole series of problems in that the data that we had was very out of date and preceded the current modern standards. So we had to go through and fix up all those bits and pieces. And over the last few years, we've been going through doing each one of those systematically. First one we've done, and this is the last thing that we've actually accomplished, which is kind of crazy, is we worked out how the drug works. We actually discovered how the drug works. And if you'd asked me at the beginning, when I first joined the company many years ago, how this drug would work, would I have guessed this pathway? And I would have never in a million years. Am I happy it's this particular pathway? And Mike will talk a lot more about it. So I couldn't ask for a more exciting and high value opportunity. Obviously, one of the problems we had is an old drug. It was originally first discovered in 1976, so it's coming up to its 50th birthday this year. There was no IP over the drug. So we've actually been able to rediscover composition of matter opportunities that will allow us to repatent the molecule as though it was just invented last week. That's an amazing opportunity to take with a drug so old. We also had to go back through and systematically collect all the data that requires and allows you to run clinical trials, so IND enabling. So we had to come up with all the non-clinical data package that goes with that, and that involved a huge number of studies and a large amount of money. We had to come up with a formulation that would actually be practical in a modern environment, which we did in the form of RC220, and we then had to actually figure out how to manufacture that at scale. Finally, this enabled us to begin the process of actually working out how do we actually optimize the clinical trial. So we now know how the drug works, we now have an optimal formulation, we now have IP, we now have all the background information preclinically that you need. We now can have the opportunity of actually going in and designing the best trials. So the original developers of the drug, Ledley, they had no idea. They just threw the drug at a cancer and saw if it worked or didn't work. They had no idea why AML works so well, and you'll hear today with Anupa will explain to you why it works so well for AML. We've got three major opportunities: CPACS, Harness 1 and AML. The team will talk to you about this, so I won't spend any time on this, but they are amazing opportunities that have enormous commercial potential and clinical potential. They have the potential to completely change the lives of thousands of patients, and this is the reason why we go to the hassle of actually struggling through and trying to bring a drug to market. And with that, I will hand it over to Mike, and he is going to give you so much science that you'll never want to hear the word science ever again. Thank you.
Right, thank you, Daniel. I'm Professor Mike Kelso. I'm the Vice President of Research at Racura Oncology. I oversee the preclinical team in charge of all the research we do, and I'm going to present much of that to you today. When I joined the company in 2021, we were a very small team. There was only 4 or 5 of us at the time. And since then, we've grown to 17 today. And I'm emphasising this because it's really been the science that's driven that growth, understanding of the drug, and it's got us to where we are today. So really that is our philosophy at Racura Oncology, is we let the science guide the direction, and in pharma, that's the only pathway to success. Before I joined the company, I was a research professor of medicinal chemistry at the University of Wollongong. I joined that university in 2008 as a postdoc and then rose through the ranks. Before that, I had got a PhD from the University of Queensland Institute for Molecular Bioscience. Did a postdoc in the Basque Country in Spain, followed by Scripps in San Diego in medicinal chemistry, where I worked with Johnson & Johnson Pharmaceutical R&D. So I'm a career scientist, career medicinal chemist. I love science, have a passion for it. I was enticed out of academia by the lure of becoming closer to where you can really make a difference to patients, and that's in industry. And having joined Racura and working with bisantrene, literally getting to live out that dream. So just a bit of a brief walk through the agenda here. So we've got a few items, as you can see in the list. I'm going to give you a brief introduction to G quadruplexes, talk about G quadruplexes as a drug target, and I'm going to do that through showing you a video that we've prepared of Professor Lawrence Hurley, who's a scientific advisory board member for the company. I'll walk through some important information about MYC as a cancer driver. This is our important target. Talk to you about bisantrene's mechanism of action, which is targeting MYC through action on G quadruplexes, and going to give you a lot of the science then. So what we've produced at Racura that supports all of these mechanism of action findings. And finally, I'm going to give you a run through of how this science and understanding of the mechanism of action of the drug has actually helped to guide us to where we are today in choosing our 3 clinical programs. Right, so shown on this slide is our wonderful drug, bisantrene. Most of you would know that we made some discoveries around photoisomerism. That's why we write it like this with those 2 E's. I won't have time to talk to you about that today. But everyone here knows that this is an old molecule discovered in the late 70s, clinical development in the 80s through Lederle Laboratories. At that time, all the preclinical studies were pointing to mechanism of action through binding to DNA, but that was pretty much all that was really understood. Due to limitations at the time in understanding of the science, molecular science, the instrumentation and experimental techniques of the day. So what we've come to understand through the work we've done over the last few years is that the mechanism of action is more complex than that. It's actually, bisantrene is a more targeted drug, where it targets these G quadruplex DNA structures, and through doing so, it leads to silencing of this really important oncogene called MYC. So just to help explain, just trying to get the laser pointer, doesn't work, so I'll have to do it here. Just 1 second, sorry. Well, we'll get a laser pointer here. So shown here on the left is DNA, as most people know it. It's a double helical structure, 2 strands of DNA wind around each other like this. But that form of DNA is not the only form. Another form that exists is called G quadruplex, and this is where 4 strands of DNA all fold around each other like this. And in part of that structure, say for example across the top here, this flat region, we can indicate with these chemical structures here where we have 4 guanine bases linked together through hydrogen bonds shown here and a metal ion in the middle. That's usually a potassium ion. And that structure is known as a G tetrad. So in this G quadruplex DNA, if you follow the sequence along here, you can see GGG, comes down here, GGG, etc., and you can see in this sequence of the DNA, you see how these G's stick together, and that allows the folding into this type of structure called a G quadruplex. And that's important to understand because this is how our drug is acting, by binding to one of these types of structures. So at this point, what I'm going to do is show you a video prepared of Professor Lawrence Hurley, who's Scientific Advisory Board member for us at Racura. He is really one of the founding fathers of G quadruplex DNA biology, and particularly around targeting drugs towards these molecules. So he's going to give you a bit of a run through and provide some of the historical context around this drug target.
It's a pleasure to give this talk on DNA G quadruplexes as new molecular targets for cancer, from conception in 1989 to bisantrene in 2026. My background is that I got my undergraduate degree from Bath University in 1967, and then my PhD from Purdue in 1970. And my overall objective for my career has been the bench-to-bedside translational science in oncology. And importantly and significantly, my father died of pancreatic cancer in 1975, which really spurred me onto this career. So the central idea of targeting G quadruplexes rather than duplex DNA originated from an article that I wrote as a perspective for Journal of Medicinal Chemistry in 1989, describing these 4-stranded structures and saying that these structures, because they were more complex than duplex DNA, will be potential receptors for selective drug action. Critical to my translational career has been Dan Van Hoff. And Dan Van Hoff I first met in 1980, and we've worked together since that time, and we have brought 4 drugs from bench to bedside, and 2 of those drugs, both quadruplex drugs, are FDA approved. Dan Van Hoff is now on the clinical advisory board of Racura, and was actually involved in the original development of bisantrene back in the 1980s, before it was known that they were quadruplex interactive drugs. The first paper that was really critical in laying the foundation for quadruplexes as drug targets for modulating gene expression, we published in PNAS in 2002. And first of all, we showed that a G quadruplex was in the nuclease hypersensitive element, which is the regulatory part of the c-Myc promoter. And then we were able to show, using 2 tool compounds, one TMP5P4, which is active and binds the quadruplexes, and TMPYP2, which is inactive and doesn't bind to quadruplexes. and clearly were able to show that TMP-YP4 modulated gene expression while TMP-YP2 did not. Importantly, in further work, we then showed that nucleolin bound to the G quadruplex, and this silenced transcription for c-MYC gene. And if a drug stabilizes the G quadruplex, it reinforces the action of nucleolin and turns off gene expression. Our first endeavor in the translational area in this quadruplex as targets for drug design started in Arizona and my lab there, and we converted a gyrase inhibitor that had good-like properties into a quadruplex interactive compound in my lab. And we then licensed this technology to Silene Pharmaceuticals, who then made a further 1,000 or so compounds to optimize the selectivity for G quadruplex over duplex DNA. From this program came 2 clinical candidates, the first one Corfloxen and the second one CX5461, that is now in phase 2 clinical trials for breast, ovarian, and also is going into clinical trials for pancreatic cancer. The first patient that was treated with Corfloxen was in 2005 in San Antonio, and Dan Van Hoff arranged for me to be at the bedside of the first patient to receive a quadruplex targeted drug. And this is a picture of me in the black coat with the patient. And what is important now is that bisantrene, which is a Racura Oncology drug, has now shown to be a quadruplex interactive compound, and it shows much greater selectivity and efficacy than other DNA reactive drugs because it binds to quadruplexes, and particularly to c-MYC, where it lowers the expression of this gene. It doesn't suffer from the adverse cardiovascular effects that are commonly found with duplex targeted compounds. It has multiple applications in cancer, is active in both solid and liquid tumors. And one example is it reverses the drug resistance in non-cell small cell lung cancer, where overexpression of c-MYC is a problem. So now the opportunity that I am involved with, that is using Racura's oncology drug to target cancer, is really a very exciting opportunity in my 50-year career. And thank you very much for listening.
So we thank Professor Hurley for his presentation there. We're really quite fortunate to be able to access his vast, really encyclopedic knowledge of G4 targeting drugs. So you heard Lawrence mention this MYC protein. And so why is MYC important? Well, MYC is what we call a transcription factor. In fact, it's a master regulator, meaning that it is in charge of controlling the downstream expression of many, many genes. And many of those genes are involved in proliferation and survival of cells. And of course, that's what cancer is. It's a disease of uncontrolled proliferation, survival of cells. So what we know is from the literature shown on this slide here that there are all of these different cancer types show these highly increased percentages of MYC overexpression. So MYC is really centrally involved in all of these different cancer types. For that reason, MYC has been considered for a very long time the holy grail of oncology drug targets. However, due to its properties, its molecular properties, the way it functions in cells, it is a really, really difficult drug target to hit. You can't just easily design molecules that bind to it and knock out its function. So MYC targeting drugs have really proven to be elusive. What I'm going to show you today is that, I'm going to pose this question, is targeting MYC via G-quadruplex the solution to that problem? And I think you'll agree by the end of this talk that yes, it is. And the molecule for the job is bisantrene. So to help you understand this mechanism of action, we've prepared a video. So I'll play this video, and after the video, I'll walk you through it again to try to help you understand a few of the details.
MYC is a master regulator of cellular proliferation and survival. When MYC levels are elevated, genes that promote cancer cell growth, proliferation and survival are switched on. MYC levels are abnormally increased in most cancers, as shown here. Let's zoom into a normal cell to see how MYC is regulated. Under normal conditions, RNA polymerase is recruited to the promoter region of the MYC gene, producing MYC mRNA, which is then translated into MYC protein. The MYC gene is tightly regulated by the transient formation of a three-dimensional DNA structure called a G-quadruplex. This structure acts as a brake on transcription by stopping recruitment of RNA polymerase. So, what happens in cancer? In a cancer cell, MYC transcription is overactive, leading to accumulation of MYC protein. This can result in a highly aggressive cancer that can become resistant to standard therapies. Racura Oncology's bisantrene has been shown in preclinical studies to bind and stabilize this normally unstable G-quadruplex. By locking this structure in place, bisantrene is believed to silence MYC transcription, evidenced through suppressed MYC mRNA levels in our preclinical studies. Because the MYC protein is short-lived and degrades quickly in cells, once production stops, MYC protein levels can rapidly decline, removing a key signal on which cancer cells rely for growth, proliferation and survival, and may resensitize cancers to standard therapies. This mechanism represents a novel therapeutic approach being investigated by Racura Oncology to target MYC-driven cancers with the potential to improve patient outcomes.
Okay, so just to go through that video again in a bit more detail. Shown here is the cell nucleus of the cell, and you can see this gene here, MYC gene, and you can see the promoter region indicated there. So as you saw in the video, the RNA polymerase comes along, generates a strand of RNA that gets translated into MYC. So that's the situation in a normal cell, and also what happens is as that piece of single-stranded DNA gets revealed, it has this ability to fold into this G-quadruplex structure. And as it does so, it shuts down the ability of RNA polymerase to be recruited to the DNA. And that shuts off further downstream expression of this MYC gene, so you can't make the protein anymore. But that G-quadruplex structure is actually quite unstable, can revert back to this form, and when it does so, you can restart the transcription and translation of the MYC protein. And in cancer, as we saw in the video, that leads to production of lots of MYC, and then MYC drives all of those downstream processes that really drive the cancer. So what happens with bisantrene comes along, binds to that relatively unstable G-quadruplex and stabilises its structure. And by doing so, it's able to shut off this downstream MYC transcription and effectively stop production of the MYC protein. So that's our putative mechanism of how bisantrene works, different to the old mechanism that was thought of in the past, where it just bound to DNA, effectively gummed up the DNA and stopped it from functioning. So what evidence have we acquired to support this mechanism of action? Well, the first evidence we've generated, we needed to show that bisantrene actually physically binds to the MYC G-quadruplex structure. And so this experiment here is called a 1 1H or hydrogen nuclear magnetic resonance experiment, where what we do is we take a molecule of a MIC G quadruplex in solution, and we titrate in a solution containing bisantrene. What I've shown over here is that G tetrad structure that I was showing you earlier with the 4 guanines, and I'm highlighting in red 4 of the hydrogen atoms that are present on each of those guanines. So remember, there's 4 of these tetrads in a G quadruplex like this. That means that there's 12 of these hydrogen atoms on the G quadruplex. And in the 1H NMR spectrum, we see these 12 peaks for those 12 guanine protons. So that's in MIC when there's no bisantrene bound. When we add exactly the same number of molecules of bisantrene to that G quadruplex, which we call 1 equivalent or 1 molar equivalent in chemical speak, we see that all those peaks disappear, and that's because there's multiple species starting to form here. There's bisantrene coming on, touching it, falling off again in multiple different ways, and that makes all of these signals disappear. However, when we get to 2 molar equivalents, so that's exactly twice as many bisantrene molecules as G4 molecules present, we see all of these signals reappear, and they appear in new positions, telling us that their chemical environment is now different, and that's telling us that 2 molar equivalents of bisantrene are now bound to this G4 molecule. So that's our first piece of convincing evidence that we're actually binding bisantrene to the MIC-G4. The next thing we want to know is where is it binding? So we know 2 molecules are binding, so whereabouts on the MIC-G4 are they binding? And there's numerous ways you can do this. Some structural biology techniques, which we're looking into. But another way is to do it in a computer, where you basically build a G quadruplex molecule, the MIC-G4, in the computer, and you put in a couple of bisantrenes and you heat this system up, effectively giving all the atoms and bonds energy. And you're looking to see whether your bisantrenes come and bind to the MIC-G4 naturally, because they've got an affinity for the molecule, and exactly where do they want to bind. So if I play this simulation, you see the bisantrene molecules, one comes onto the bottom and stays there, one comes onto the side loop. Up the top you can see it's wiggling around as the simulation proceeds. It eventually comes off that spot and ends up sitting on the top and finds the lowest energy, highest affinity position there. So that, we believe, is probably the most populated structure where 2 bisantrene molecules are bound to the MIC-G4. This is a theoretical study, so as I said, it's in a computer, so you need to do some further follow-up work to prove that, and we've got those studies happening at the moment. Right, so now we know 2 molecules of bisantrene bind. We've got a model of where they bind. The other key piece of critical information is how tightly do they bind, and you want them to bind as tightly as possible, because if they're not very tightly bound, they fall off and then MIC just does what it does again. So we use this technique called surface plasmon resonance spectroscopy. What you do is you take a chip which has a surface, you attach chemically some MIC-G4 molecules to it, and you flow across that surface some EE bisantrene solution, and you shine a light source onto that surface. It gets reflected and picked up by a detector. When those bisantrene molecules bind to the MIC-G4, they alter the refractive index of the solution and the surface here, and you get a change in the signal you detect. And so what you do then is you titrate in increasing amounts of bisantrene in that experiment, starting off low concentrations. You detect a signal. So as you increase more and more bisantrene, you see the bigger and bigger signal here. And so what you do then is you apply some mathematical formulas, and you generate what's called a dissociation constant. And so that number, KD, tells you how strongly your bisantrene molecules are bound, and you want that number to be as small as possible. So here it's 208 by 10 to the minus 9 molar, which is a very small number, and that tells you that you're right in the range of where you need your molecule to be binding tightly to have this physiological effect on the MIC expression that you want to see. Another experiment that we wanted to do was to show that not only does it bind to the MIC-G4 and we need to know that it stabilises the structure. If you remember from the video, when it binds, it stabilises it, it stops it from transferring back to the form that's then again able to transcribe to MYC. So we do this by circular dichroism spectroscopy. This is an experiment where you take a solution of MYC-G4, shown here, and you shine circularly polarised light through that solution. You don't need to know what that is. This structure will have an effect on that light, which you can read out as a signal here. And as you increase the temperature, that MYC-G4 structure starts to unfold, or denature is the term we use, and ends up becoming a linear sequence of DNA just like that. So the amount of energy required to do that unfolding is read out by the 50% inflection point here, and so that temperature is 33 degrees. If our bisantrene is binding to that MYC-G4, we expect that to stabilise the structure and increase the temperature required to melt that complex. Right, and that's exactly what we see in the green over here. We see that the 50% unfolding temperature has moved from 33 to 68. Okay, so we know that we're stabilising the structure of that MYC-G4. Okay, all of that's all well and good, but that's all in the test tube. There's nothing, no cells there, no tissue, no anything. So what you need to know is, does all this read out as an effect in cells that you want to see? And the effect that we want to see is a reduction in the production of MYC RNA, because that then gets transcribed and translated into protein. So what we've done here is we've taken 3 cell lines. One is a breast cancer and 2 non-small cell lung cancer lines. One's resistant to this drug osimertinib, which I'll talk about more in a minute. And you can see that as we increase the concentration from left to right of bisantrene, in each of these 3 cell lines, the MYC expression levels are all going down. And we take the 50% number from over here, and we read that out as an EC50. And we can see that the EC50s here are all in this nanomolar range, which is, again, where you want it to be. You want that to be occurring at very low concentrations of bisantrene, so that you know that in cells it's going to do that, and you know that at reasonable doses of your drug, you're going to be able to produce those levels in cells. So that's a simple way of looking at it. Here's a cell nucleus producing lots of mRNA from MYC. Bisantrene comes along, and we've reduced those mRNA molecules. All right, so this is just a quick summary of all the things I've just shown you. We showed firstly that MYC binds to, bisantrene binds to MYC in a 2-to-1 ratio. We found where it bound by molecular dynamics. We showed how tightly it bound by surface plasmon resonance. We showed that it stabilised the G4 through circular dichroism. And then the clincher was this one here, gene expression analysis, where we can show that we can actually stop MYC getting produced in these cancer cells. All right, so what you've been hearing so far is about MYC and its role in driving cancer progression. But another property of MYC, it seems, is that it is also responsible for production of what we call acquired resistance to targeted therapies. So targeted therapies have really been revolutionary in lots of areas of cancer over the last 20 years. And they work well quite often for a while, sometimes some years, but eventually many patients will suffer acquired resistance where the drugs just stop working, and then they've got a very poor prognosis after that. And then if you look in the literature, you'll find many papers like this that really support this idea that MYC is a key driver of acquired resistance to targeted therapies. So along with that knowledge and this knowledge, that over the last few years we've done a number of preclinical studies, some of which many of our shareholders will have seen because we've announced them to the ASX, where we're looking at things like tyrosine kinase inhibitors in clear cell renal cell carcinoma. We've also looked at BRAF and MEK inhibitors in melanoma, a couple other clear cell renal cell carcinoma compounds, and also in AML. And what we've noted is we see really good synergy with these targeted agents and bisantrene. These pictures here I'm showing you, don't have to understand what they mean, just look at the pluses. The pluses indicate where you see synergy between the two drugs, so you can see that it really is broad synergy we see across multiple different cancer types, multiple different targeted therapies for those cancers. So this made us, all this information about MYC being involved in targeted therapy resistance, all the work we've done on targeted therapy synergy, made us ask this really key question: which unmet needs in oncology are MYC driven and have the best clinical and commercial opportunity for the company? And we answered that question by saying it looks like acquired resistance to osimertinib in EGFR, that stands for epidermal growth factor receptor, mutant non-small cell lung cancer. And one of the key papers that made us, convinced us this was our area to get into was this one published by Professor Shiyong Sun. He's at Emory University. Many of you would know we now collaborate with Shiyong on this very program. So this paper they published really pinpoints the role of MYC as a driver of acquired resistance in non-small cell lung cancer when you treat with osimertinib. All right, so what evidence have we got now to support the potential effectiveness of e-bisantrene and RC220 in non-small cell lung cancer? Before I get into the actual evidence, let me just give you a quick run through explaining how this all works and why we think bisantrene is going to work here. So shown here is a normal cell. In the cell membrane, have this receptor called EGFR. What happens is EGFR receives its endogenous ligand, activates all these intracellular pathways, and many of these pathways converge on this key protein MYC. MYC then does what it does, it binds to DNA and activates the transcription of many different downstream target genes, causing cell proliferation, survival effects. And they're all, that's all normal in most cases, but in cancer, what happens is, in this type of cancer, that receptor becomes mutated, and it becomes mutated to a point where it becomes what we say is constitutively active, where it's just on all the time, signalling all the time, all these pathways, making MYC active, and then you get this tumour cell proliferation uncontrolled, and that's cancer. So then this cancer gets treated with osimertinib. It effectively shuts down this receptor by binding to it here. All this downstream pathway is inactivated, and we don't have cancer growth anymore. And that works very, very well for a period of time. But what happens with time is you can get all of these additional new pathways start to get activated. These are called bypass resistance pathways, and that reactivates all this system, and you end up with osimertinib resistance, and then the patients aren't going to last very long after that without another intervention. So where does bisantrene, RC220, fit into this? Well, you're hearing now that it has an effect on this MYC, so we expect it to be able to shut down this reactivation of all these pathways at this downstream level where MYC is involved, and that's going to have this impact on taming down resistance to osimertinib. Right, so shown on this slide is an experiment which is called RNA-seq or sequencing analysis. Here we've taken two cell lines, PC9 and NCI-820. These are two non-small cell lung cancer cell lines. We treat them with e-bisantrene, and then we measure its effects on all of the genes in the cell. So all of the dots in red indicate genes that become upregulated, so there's more of them in the cell after bisantrene treatment. All the ones in blue are the genes that become downregulated or suppressed by bisantrene treatment. And you can see that in both of these cell lines, there's really a huge number of genes that get affected by bisantrene treatment. After we've done that analysis, what we do then is a follow-up analysis called a pathway analysis. This is where we look at the pathways involved in all of those genes that get changed. And you can do this through bioinformatics using the computer. So all the pathways on the right here, these are the upregulated ones. All the ones on the left here are downregulated. And we can see that in this PC9 cell line on the left, that the two most downregulated pathways involve MYC. And we can see over here in the osimertinib resistant cell line that the only pathway downregulated by bisantrene is MYC. So clearly MYC we know it's heavily involved in resistance in these cell lines, lung cancer, and we now know that bisantrene is able to downregulate MYC in these cells. The next piece of really great data that we've acquired are these 4 mouse model studies shown on this slide here. And this work was done by Professor Xiong Sun's lab at Emory. So what we do is we grow tumours in mice, EGFR, non-small cell lung cancers. We measure the size of those tumours over time, and we treat them either with vehicle or these treatments over here. And you can see that in each of these lines, osimertinib in blue is really not doing much at all. So these are all osimertinib-resistant lung cancer cell lines. And we can see that the combination in red is really having quite a profound impact here. We're really pushing down these tumours and preventing their growth in these models. So that's really encouraging to see. In this model, we actually use PC9 cells, which are sensitive to osimertinib. So it's a mouse model again. Each of these lines represents a single mouse and the size of its tumour as it grows with time. And so you can see the vehicle-treated mice, their tumours are growing really rapidly, and they don't last beyond about 15, 16 days or so. When we treat with osimertinib, all of the 10 mice in that cohort in blue, you can see their tumours all regress. So it works really well. And then if we stop treating, all the tumours start growing again at this rate here. You can see with the combo, though, osimertinib and bisantrene, we get this really good repression, right up, even better than osimertinib alone. And then we stop treatment at day 18, and we get regrowth of the tumours. And we can see that the rate of regrowth is actually being slowed greatly compared to osimertinib alone. And then if you plot all that data on what we call a Kaplan-Meier survival curve, you see in blue osimertinib treatment. You can see each of these decreases in the line here represents death of a mice, and we can see that the mice treated with the combo have a really increased survival benefit in this particular model. That's the kind of curve you want to see in a clinical trial when you're looking at overall survival, so people untreated with RC220 versus those that get it. All right, so moving now away from non-small cell lung cancer to our second clinical program that we have in AML. And of course, the theme of this mechanism talk has been that the work we've done, the learnings that we've made, have helped to guide us choose these clinical programs. And you'll see now that this knowledge really does support our AML program. So this is a picture you saw before, all the different cancer types which are showing MYC being upregulated. But you see down here leukemia, 20%, you think, well, that's not a very good number. Why would we choose AML? Well, that's only a small part of the story because AML is many different types of blood cancers shown here. And you can see this one at the top, AML, 90% of AMLs are known to be driven by MYC. So it's clearly one that sits right in the slot for where we want to be looking clinically. Here's another RNA sequencing study. This time we're using MOLM13 AML cells. So we're looking at which genes get upregulated and downregulated when you treat them, those cells with EU bisantrene. All the upregulated ones are shown here on red on the right and downregulated in blue. We've cheated here though, we've skipped a step, and what we're showing here is actually the pathways that are up or down regulated rather than just the genes. So you can see here that MYC pathway is the top downregulated pathway in these AML cells. We know that because the further to the left and the higher up they are, that's the strongest signal in this RNA pathway analysis. So MYC is clearly involved in AML, and bisantrene is clearly having a profound effect on suppressing MYC in this experiment here. And when we do a couple of mouse models in AML, what we find is that we got a MOLM13 model and a patient-derived xenograft model over here. These are the Kaplan-Meier survival curves. Vehicle-treated over here, these mice all died before 20 days. You get a small effect at a low dose of bisantrene and a much greater survival effect at a higher dose. And that's exactly what you want to see. You want to see this dose dependency in the effect of your drug to tell you that it's really doing what you think it's doing. And we see the same effect, even a bigger survival effect with the higher dose of bisantrene over here. All right, so that's a bit of data for AML, and I'm just going to finish off now by talking very briefly about our CPACS program. How does this MYC G quadruplex DNA binding story fit into this program? Does it fit at all? Shown here on the left is data that has been released by Racura previously. This is data that was presented by Daniel at ESMO on his poster last year, showing that when you treat cardiomyocytes cells with doxorubicin, what you get is a really strong signal of double-strand DNA breaks. And we think, many people think and agree with this, that double-strand DNA breaks in cardiomyocytes is the reason for cardiotoxicity of doxorubicin. So when we do this experiment, we add more and more bisantrene, we see that the number of double-strand DNA breaks is going down. And so we're very confident the reason bisantrene is able to protect the heart against doxorubicin cardiotoxicity is because of this ability to reduce the number of double-strand breaks in the cardiomyocytes. So then that begs the question, how does it do that? How does it stop those double-strand breaks from forming? And the clue to that answer comes from this paper that was published in 2021, in 2022, Molecular Cell, that talks about MYC and its role in assembling something called a topoisome, which involves these topoisomerase enzymes 1 and 2. Topoisomerases are enzymes that cut DNA, either make double-strand cuts or single-strand cuts. So how could this happen then with this topoisome and bisantrene? Well, here's a cardiac cell represented on this slide. If we treat that cell with doxorubicin, one of the things we know about cardiac cells is that causes an upregulation of MYC. So you get more MYC produced in these cells. Right, that paper that I just showed you, it talks about the assembly of this structure, which is called a topoisome, involving topoisomerase 2, topoisomerase 1, another protein called MAX and MYC. And this assembly then comes along and it binds to DNA. Topoisomerase 2 makes a double-strand cut in the DNA. Normally that gets healed by the processes of this topoisome. However, in the presence of doxorubicin, that actually binds to topoisomerase 2 and poisons it and prevents its ability to repair those double-strand breaks. And that results in cardiotoxicity. So then in the presence of bisantrene comes along, we know it's going to have an impact on that MYC levels in those cells, going to suppress them, and that's going to mean that less MYC around, less topoisome, and therefore less double-strand DNA breaks. And that means we've got cardioprotection. So this is all a hypothesis at the moment based on what I've shown you, and we've got lots of work going on in the lab and elsewhere looking to consolidate that hypothesis with data. All right, so I know I'm just over time, so I'll be brief here. Just in summary, I've given you the new mechanism of action of bisantrene, which focuses on the drug bisantrene binding to the MYC G quadruplex. So that's located in the promoter region of the MYC gene, has an effect on MYC expression in cancer cells, and also inhibits MYC downstream target genes. So I hope I've convinced you now why all of this knowledge around this mechanism of action has helped us in our delivery of these three clinical programs that we're now undertaking. So just the final message here is we've done obviously a huge amount of work, including work done by our in-house Wollongong University scientists shown down the bottom here, others over up the top here. David's a new recruit, he's in charge of CMC. But lots of us have done lots of work outside the company as well through various vendors where we contract studies, generate all the data for us and bring it in-house. So that's where I'll leave it. Thanks very much for your attention and happy to take some quick questions. We do have another question session right at the very end, so in the interest of time, we'll probably only take one or two at this stage.
bisantrene, they noticed an effect on meat, but they thought it was a off-target effect. What was it that enabled Racura to come to the correct conclusion? Is it that methods are now available which weren't available to City of Hope, or is it that you're better scientists?
I think we all know the answer to that one. I'll just say that, yeah, I know the experiment you're referring to in that paper, which shows suppression of MYC by bisantrene, and they attributed that to inhibition of the enzyme FTO, and then that was a downstream effect of that inhibition. We've done enough experiments to know that's not what's going on. We know that it's more what I've shown you today. That's the explanation for that result in that paper.
Sorry, maybe a segue to that same question and asked a different way. You put a lot of slides on affinity, but you didn't put anything on selectivity. So how selective is it? Because targeting these quadruplexes has been decades, as far as I know, and there's hundreds of thousands in the genome. So how do you know it's targeting the one you want? I think to that same question.
Yeah, no,
selectivity.
That's a great question, and you obviously know the field. The selectivity is a great thing to consider. We know that it does bind to DNA as well, double-stranded DNA. You know, you look in the past, there's evidence for that, for sure. But the thing that tells us that it really is focusing on the G quadruplex, particularly the MYC G quadruplex, is that signal in the expression where we see the dose-dependent drop in the MYC expression level, and you know it's clearly a signal that we're hitting that particular G quadruplex with all the other evidence that we've acquired. Sure, it's probably hitting some other G quadruplexes as well. We don't have a magic bullet G quadruplex binder here, but it's doing the key thing that you need it to do, and that's to downregulate that MYC.
Okay, Mike, I think we've got it. All right, thank you, Mike. That was great. Thank you very much. So that's the heavy science out of the way. So you can all breathe a sigh of relief. We're going to be heading into the clinical section. But just to really say, emphasise why it's so important to understand this mechanism of action, particularly on a big target like MYC. Convincing pharma is always really, really challenging. And so these orthogonal approaches look at using different techniques, and they're all pointing at MYC. As Mike said, probably having actions elsewhere as well, and we're going to be looking into that, but clearly the heavy lifting is being done by sciencing of MYC. So I'll move on to the clinical programs now, and just, this is our portfolio. We've got the 3 programs there that Mike has described, including at the top AML going into phase 3. 3 very, very different risk profiles. AML, we know it works. We know that's a MYC-driven cancer. And then we're asking 2 different questions in the other studies. One is, does it work to overcome resistance? That's the Harness 1 lung cancer trial. And then CPACS, can we protect those cardiomyocytes, the muscle cells in the heart, from damage caused by doxorubicin? So very different risk factors. How that plays out, do they all 3 work? I hope so. If 1 or 2 work out, we're going to be very happy as well. The other thing, just to emphasise, and the teams in the room, you can spot them by their mugshots here. We've really grown the clinical team over the past 12, 18 months. Some excellent people, including some who are going to be talking this afternoon. But we really are bringing the skills in-house to deliver and execute on these clinical trial programs. They require a lot of attention, and you need to know what you're doing. This is no place for gifted amateurs. So just to emphasise the clinical team, and Anupa is heading up AML, Rodney's heading up the Harness study, and Marinella is heading up the CPACS, the Cardioprotection anti-cancer study. So with that, I will hand over, first of all, to Associate Professor Erin Howden, who is Professor of Exercise Physiology at the University of New South Wales, and then she'll be handing over to Marinella Messina, who's, as I said, head of the CPACS trial and Racura's Vice President of Clinical. So welcome, and thank you for being here.
Thank you, Peter, and thank you for the invitation to present at the inaugural Racura seminar. So, yes, my talk is going to be very different to Mike's. So, Okay, so my background, I'm a clinical researcher. I'm an exercise physiologist by original training, but my research is really focused on cardiovascular physiology. And I put these two images up because I think it kind of encapsulates my research program. I use exercise to try and understand physiology, in particular how the body uses or is able to create energy to do movement. And we do that by studying what's called the oxygen utilization cascade and we use that to understand mechanisms of exercise intolerance, especially relevant for people with heart failure or individuals who are at risk of heart failure, which comes into this CPACS program. The other aspect of my research is on the adaptive capability of the cardiovascular system. So we are interested in the activity paradigm across the spectrum. So that's from the most inactive individuals to very highly trained athletes. So what has all of this got to do with patients with cancer? So we know that cancer is really an accelerated cardiometabolic aging phenotype. So if we think about aging across the lifespan, basically each year of our life we are losing our functional capacity or cardiac reserve. If you are unfortunate enough to be diagnosed with a cancer and need to receive a potentially cardiotoxic treatment like doxorubicin, it can really accelerate that cardiometabolic aging process. And so we lose cardiac reserve when we're exposed to these treatments, and the consequence of that is that you're at an increased risk of developing heart failure. So a lot of my research is focusing on trying to prevent that, trying to identify people who are most at risk, identifying strategies to try and reduce the likelihood of that. Now, the heart is obviously very important. There's a whole field of cardio-oncology now that's focused on protecting hearts of cancer survivors, but it's not just the heart that's affected. So we have a number of contributing factors that increase the risk of cardiotoxicity when you're exposed to these chemotherapies, but the impacts of these therapies are not just on the heart, and we've got evidence to suggest that there's impacts, well, not just us, but other groups have shown that it impacts pulmonary function, vascular function, the circulation, the skeletal muscle, and this all kind of encapsulates and reduces the cardiac reserve of the individual, and that's why we've got to be worried about heart failure, heart disease in these patients. So the impacts of anti-cancer therapy are not just at the time of therapy, they are long-lasting. So this is a systematic review that was published just last year in JACC Cardio Oncology, and they looked at how cardiorespiratory fitness, or VO2 peak, was impacted by patients who were exposed to anthracyclines chemotherapies. And you can see that across the spectrum of the treatment, following their treatments with systemic therapies, that their reduction in fitness, or VO2 peak, relative to age-matched controls was progressively worsened as they move further into survivorship. So we've also looked at this, and some of you may have been familiar with this study. I think it's been spoken about previously, where we looked at this prospectively in patients who are initiating anti-cancer therapy in Melbourne at the Baker Heart and Diabetes Institute. And this was a mixed group of individuals. Some patients were receiving anthracyclines, some were having allogeneic stem cell transplants, some patients were receiving treatment for prostate cancer. But they all had a follow-up cardiopulmonary exercise test about 3 months after initiating their therapy. And you can see the results here. So we saw this is their VO2 peak at baseline. You can see that in basically all of these participants had dropped during that 3-month period. Now, in the patients who were treated more aggressively, so those with breast cancer or those receiving an allogeneic stem cell transplant, we saw significantly more marked reductions in VO2 peak. What we did in this study as well is we looked at how these clinically significant drops in VO2 peak overlapped with other traditional markers of cardiotoxicity. And so we can use echocardiogram echocardiography to look at changes in ejection fraction or global longitudinal strain, and they're probably two of the most commonly used techniques available. We also measured biomarkers, and there are threshold cutoffs for all of this to identify if you meet criteria for cardiotoxicity. And what we wanted to see with this figure is just how well all these cutoffs agreed with each other. And I think you can see from the slide that there is actually pretty poor agreement. Now, others have also looked at the relationship between exercise capacity or VO2 peak and risk of cardiotoxicity. And in this study from Canada, they actually did show that a lower VO2 peak was associated with poorer cardiac function. So the people with the lower VO2 here did have poorer ejection fraction, and they have, and they also have reduced global longitudinal strain. So it suggests it's not conclusive. There is clearly a role for, or there is an impact of these treatments on the heart, but it doesn't explain the full story when we think about the overall effects. But we do know that this is important. So if you have a reduced cardiorespiratory fitness as a cancer survivor, you're at an increased risk of all-cause and cardiovascular specific mortality. So for each 1 MET increase, which is a unit that we use to measure cardiorespiratory fitness, for each 1 MET increase in fitness, you had a decrease of 26, 14 and 25% in all-cause cardiovascular and cancer mortality. So it plays a role. If your fitness is higher after treatment, your survival outcomes are better. But what does this mean on a day-to-day basis for our cancer survivors? So you're all sitting very relaxed in your chairs right there, listening to me speak about VO2. At the moment, you're probably using about what we'd say 3.5 to 4 mils per kilogram per minute of oxygen just to be at rest, to be seated. So if you've got an average VO2, you're good, okay? But our cancer patients, after treatment, their VO2s are in this blue range over here, so between 15 and 20 generally. Now, if you look at these general activities of daily living, for these individuals, performing these activities becomes a very significant workout. And we have this term called an anaerobic threshold, a lactate threshold. There's lots of different ways we describe this in the literature, but essentially it's the time when you're exercising when it starts to get really hard, where you start to notice that you're being breathless, your heart rate's quite elevated. So for these individuals, sweeping, carrying in groceries from the car can become challenging because their fitness has been so reduced. We've seen that in breast cancer survivors, 12 months after their treatment, that their VO2 peak continues to remain impaired. It's not something that just bounces back. So we need to be thinking about how we can prevent this happening. We also see that it associates, or it's related to cardiac dysfunction, the emergence of a loss of cardiac reserve. So like we hypothesized in that original figure, we see that cardiac reserve is also impaired in these breast cancer survivors. But like I said, it's not only the heart. So if we look at across, we use that oxygen cascade model that I described in my first slide, we can look at all of these different elements that are involved in creating energy in the body and producing our VO2. And we can see that in breast cancer survivors, that it's not just simply one factor that is leading to a reduced cardiorespiratory fitness. And so we need these multifaceted interventions or preventative strategies to try and improve this for our survivors. So if we focus on preventing or correcting only one of these parameters, so in the setting of cardioprotective pharmacological strategies like ACE inhibitors, beta blockers, they really focus on cardiac protection. So if we have an effect on the heart, that's great, but it's not going to completely solve our problem. We need to be targeting multiple processes or multiple parameters to try and optimize performance. And I'm sorry I didn't get to talk when it was race, but yeah, I'm sorry. So why do we need to be worried about this issue? There's lots of, obviously, research going on in the field to try and identify ways to protect the hearts of cancer survivors. You hear more about the uses, how frequently doxorubicin is used by Marinella, but just to highlight that cancer cases globally are rapidly increasing. So this is just a paper here that was published only last week in the Lancet Oncology, looking at the rates of breast cancer incidence, looked at the projected rates of breast cancer incidence in the next 25 years. There'll be a 50% increase by 2050 in the number of women that are being diagnosed with breast cancer. And you can see here that when those cases occur are in midlife. We want to be protecting these hearts of these women so that they can maintain active lives, be able to contribute to society and not become our heart disease patients of the future. At the moment, heart disease, cardiovascular diseases are the leading causes of death globally. The rates are already astronomical. With the ageing population, the growing burden of cancer, these rates are unfortunately going to increase because we're doing a better job of helping cancer patients survive their cancers. So I guess I wanted to just leave you with that. We need more solutions to this problem because it will not be going away without better treatments at this point. So thank you.
Hello everyone, welcome to this part of the talk, and my name is Marinella Messina, and I'm, as Pete Smith has told you, I'm leading the CPACS program. And this afternoon I'm just going to take you through the phase 1 trial that really marks the start, the clinical start of the CPACS program. So thank you, Erin, for highlighting the clear fact of the role of VO2 peak as a cardiorespiratory marker that really can be used to be a comprehensive measure of the effect a drug can have if it's protecting the heart. So I'll just start first with addressing the clinical context with anthracyclines. So anthracyclines remain the cornerstone of cancer treatment. As this bar graph shows you, they still are broadly used across a range of cancer types, both solid tumours and hematological tumours. And what you can also see in this graph is that their use is particularly prominent, and as Erin has explained, in breast cancers. And this includes in the neoadjuvant right up front of their treatment pathway, as well as in metastatic settings. Okay, so the use of anthracyclines obviously is still active and key. And what this actually translates to is that globally 20 million doses of anthracyclines are administered every year. So this really highlights the clinical importance of anthracyclines still today, and also addresses the scale of the effect on patients. So what do we know about anthracyclines is that they're very highly effective, but they come with a serious side effect, and that is cardiotoxicity. So the cardiotoxicity is cumulative, it's often irreversible, and as a consequence it leads to a limit of the use doses of anthracyclines in patients. So for patients in particular that have late-stage disease, they are restricted from optimal therapy because of their lifetime limits. In addition, the actual chemotherapy damage in the heart actually works in stages, and I think Erin's sort of addressed that a little bit, and it starts from early microscopic injury to structural decline to a loss of functional capacity. So this is a continuum of damage that occurs throughout. So what the CPACS program is interested in is finding one of those solutions to the cardiac risk part. And this brings me to the phase 1 clinical trial, and what you see here is the overview of what it's looking like. So what this particular trial is designed as a 2-part integrated study, where we first start at the phase 1A dose finding part, where we need to look at the safety of the combination of RC220 with doxorubicin and look for generating the maximum tolerated dose, so we know that that's going to be safe moving forward, as well as generating early signals of efficacy. And in this case, efficacy is the fact that you can continue the anti-cancer effect, making sure we maintain that, doxorubicin, if not actually improve it, as well as protecting the heart. Once we complete this component of the study, we transition to a phase 1B, where we will look at the maximum tolerated dose combined with doxorubicin in an expanded cohort, and here we look more into the functional efficacy signals, and this will include VO2 peak as a clear measure of assessing the effect of cardioprotectant agent. So just moving on a little bit more detail at each of these studies. We'll move on to this phase 1. So in this phase 1, 15 to up to 33 patients will be enrolled, and these patients are advanced metastatic disease patients that potentially can benefit from doxorubicin treatment. This is a dose escalation study, so the doses of RC220 will increase in combination with a fixed dose of doxorubicin at 60 milligrams per metre squared. So the overall schedule of treatment you'll see here. So we also have some 2 upfront monotherapy cycles of doxorubicin and RC220. Then they follow in the repeated cycles of combination. So the 2 lead-in cycles have a couple of advantages. First of all, we use the doxorubicin upfront monotherapy to check that the patients can tolerate the 60 milligram dose of the doxorubicin. And this is particularly important in this patient population because they are advanced, they would have had earlier treatments for their cancer, and as Erin has pointed out, they probably have reduced functional capacity. So being able to tolerate the dose of doxorubicin is important to know this up front. Once they pass this tolerability stage, they can then receive a single cycle of RC220, which is really the initial assessment of their safety with this new, in this case, new formulation and new drug. And then we move on to the combination cycles. So what are our goals? Our goals in this study are, as I've mentioned before, focusing mainly on the safety and tolerability of the RC220. Also, key is to find that maximum tolerated combined dose that we can continue to pursue, as well as generating pharmacokinetic data, and that is the absorption of the drugs and how they interact with each other and their metabolism in the body. These are highly important information to understand those associations. And we'll also, in this early first study, collect some preliminary signals of the cardioprotective effect using a biomarker, a molecular biomarker, to check the actual change in the DNA to indicate cardioprotection. We're also looking at preliminary signals of anti-tumour efficacy, and this will be done by imaging. Once this is complete, all the accumulated safety data, the PK, any efficacy signals will be reviewed in an interim analysis to then progress to the phase 1B. The reason for the interim analysis is really to further refine the potential design of the phase 1B and or confirm as it exists now in a planned stage. Okay. So, phase 1B. Clearly this is going to be a key part because now we have the maximum tolerated dose to advance in an expanded cohort of patients. These patients, you'll notice these are going to be patients with solid tumours, but they're anthracycline naive, that is, they've not been exposed to doxorubicin. This provides a population that's in one way cleaner, so we can really assess the cardioprotective effects, in particular with RC220, without any interference from prior doxorubicin or other anthracycline therapies, which could have provided, could have left the patient with some pre-existing cardiac damage. So in this study, we'll use the maximum tolerated dose combined with doxorubicin in repeated cycles. And the goals here in this study are twofold areas. So we want to look at anti-cancer effects. This is also to confirm safety. We'll also look at the anti-tumour effects using imaging, evaluating hair loss. Alopecia was noticed in the past to be reduced when bisantrene was administered in the phase 3 study. So we'll be pursuing that and looking overall at their quality of life. And when it comes to the cardioprotective effects, key to the assessment for cardioprotective effects is the VO2 peak, being a more comprehensive measure of cardiovascular health, and also, as Erin has pointed out, it is potentially providing early signals of cardioprotection ahead of the more established assessments of, that Erin has also pointed out, of GLS and LVEF, or left ventricular fraction, and global longitudinal strain. Of course, we'll be looking at these heart-specific measures as complementary to these findings to provide a very comprehensive assessment of the cardiovascular response, the cardioprotection response. So, at the end, we're looking to determine two effects, a dual effect, both anti-cancer and cardioprotection, in this early preliminary stage. And that's the end of this study, and results from these studies and these findings will inform the next progression to phase 2. Thank you.
Thank you. We have run a little bit over, so maybe we can save questions to the end.
Perfect.
That's great. Thank you. Good. Now we're going to switch gear and switch to the Harness 1 trial, the non-small cell lung cancer trial. So it's my great privilege to introduce Professor Nick Pavlakis, who's Director of the Medical Oncologist at Royal North Shore Hospital, specialising in thoracic and gastrointestinal cancers. Until recently, Nick was Chair of the Thoracic Oncology Group of Australia, the peak organisation for lung cancer specialists in Australia. And after his talk, he'll hand over to Rodney Cusack, Principal Scientist at Racura, who's the project lead on the Harness 1 trial. So welcome, Nick. Thank you. I'll go around this.
Thanks very much for the opportunity to present today. It's a privilege to be part of the program. So I'm a medical oncologist. I'm a clinical academic. I run clinical trials from drug development, early phase, as well as biomarker discovery and validation. So I work at St Leonard's, but I work for Royal North Shore. And TOGA is our clinical trials group, our collaborative national clinical trials group. And the other one I work with is the GI cancer trials, which was formerly the AGITG. These are my disclosures. I'm going to talk to you primarily about lung cancer today, try to set the scene for why the Harness 1 trial is trying to address a clinical gap. So lung cancer is the leading cause of cancer-related mortality worldwide. And on the left-hand side of this curve, you'll see the number of cases that we are encountering today and where it is expected to lead over the coming years. By and large, it's still considered a smoking-related disease, but it's not exclusive. And I treat a lot of non-smoking-related lung cancer. There's environmental exposures, there's occupational diseases, there's chronic lung diseases as factors. But I'll show you how this particular disease we're going to focus on today, which is EGFR mutant lung cancer, is one that occurs also quite heavily in non-smokers. Now, lung cancer, when I see a patient, the first thing I say is that they think of it as a death sentence. And I say, look, not all lung cancer is the same. We need to look at which specific type of lung cancer you have. Now, the ratios you'll see have changed. When I started, it was 25% small cell because it was more heavy smoking populations. Now we're down to about 15% and less, and depends on which part of Sydney and demographic you're in. We're going to focus primarily on non-small cell lung cancer on the right-hand side, and the key subgroup of non-small cell lung cancer, which is adenocarcinoma. Now, this pie chart was written by one of our ex-fellows, is now working in Singapore. It demonstrates the molecular subtypes of adenocarcinoma of the lung, which is the type of lung cancer that occurs predominantly in non-smokers. Now, EGFR is the dominant molecular subtype. This is the one that's driven by specific mutations. We heard earlier about what an oncogene is. It's a driver that causes downstream repetitive signaling in a cancer. You'll see there that the big blue mark is 46% incidence, EGFR in Asian countries. In some countries, it's 50% and higher. In Caucasian populations, and the middle is the middle part of the curve demonstrates about a 16% incidence of EGFR in Caucasian populations. And that'll depend on city by city according to how much, migration there is. On the North Shore of Sydney and down in Hurstville, we see a lot of oncogene-driven lung cancer because a lot of Southeast Asian population. And then the other very smaller types I'll show you a little bit about, these are other oncogene drivers in lung cancer. I heard a little bit about EGFR. It's a transmembrane protein. It causes downstream signaling. We heard that MYC is pivotal down the bottom in propagating. It's almost like where the focus is of all the other pathways. It's a transmembrane protein, and a lot of the discovery of drugs to target it either focused on the cell surface, on the outside, using antibody therapies, but the successful ones were the small drugs that got inside the cell and blocked what's called phosphorylation inside, and that stopped the signaling. The current drug we use is osimertinib on the right. We call that a third-generation tyrosine kinase inhibitor. But the first-generation drug, gefitinib, was first discovered in the early 2000s, and the association with gefitinib with EGFR oncogene was around 2003. Now, this particular subtype of lung cancer has a high propensity for metastases. Because these are non-smokers a lot of times, or long-time ex-smokers, they're generally not part of screening programs. So these are patients who often present with symptoms. So we often see this, the characteristics of a patient with an EGFR cancer are widespread metastases because of the growth characteristics and survival characteristics that this oncogene seems to drive. Exon 19 and exon 21 are the two common types of point mutations, or the two most common types that we see. So EGFR mutations are more common in Asian patients, more common in females, more common in non-smokers, but also long-time ex-smokers and former smokers. Still a small incidence in smokers, and in adenocarcinoma. They're generally a population that presents with symptoms because they tend, as I said, not to fit into the screening populations. And even though in Australia we've started lung cancer screening on July 1 last year, this is a subgroup that won't really fit into that. In fact, the de facto screening population I tend to see are those people who have gone in for CT coronary angiograms, and we see incidental lung cancers, and there often we find this type of lung cancer there. On the left-hand side, you'll see the difference in the prevalence in Asian countries versus the Caucasian populations. The Asian countries is actually a little bit higher, up to 50%. The metastases can be in widespread parts of the body. About 20% can also occur within the brain at diagnosis. Now, this is a timeline showing the Federal Drug Authority US, in the US, the approval of therapies. In the Australian context, it mirrored it just a little bit behind. We tend to follow the US FDA by several years through our TGA and PBS process. Our main drug, and the red box covers where EGFR is. The others highlight the different oncogenes I showed you on that pie chart. We're over here at the moment. There's some new drugs being developed we call fourth generation drugs. And what we're really doing today in the EGFR space is looking at combination therapies and trying to personalise therapies. This is just a set of lungs. You can see windpipe, the right-hand bronchus there. If you think about a tumour compressing that bronchus, you can imagine the type of symptoms someone would declare: a cough, breathlessness as you're compressing some of the airway, hemoptysis is blood in the sputum, wheeze or pain. So patients will often present with symptoms. One of the key characteristics of these new generation drugs is they induce a tumour response or shrinkage. So when you reduce the tumour size, these effects of the compressive effects and infiltrative effects go away, and the patient feels better and the symptoms tend to go away. So you'll get a patient coming in with lots of symptoms, they'll go on these drugs, but as they progress, these symptoms start to reoccur or metastases will develop. And the purpose of the trial that we're talking about today is how can we delay that onset of progression before symptoms start to recur in patients. It is a universal problem that patients with metastatic disease will develop resistance to therapy. I do have patients who have survived up to 8 years with this drug, but they all eventually develop resistance. Currently there's various different approaches to try to overcome that resistance. The median progression-free survival, the time before progression is seen on an imaging analysis, either a CT or an MRI, is usually around 18 months. There doesn't seem to be much difference in the modern third-generation drugs in terms of time to development of that resistance, and some of the what we call fourth-generation drugs now don't seem to be overcoming this in the clinic. The main reason for resistance is predominantly biologic, not pharmacologic. And we heard earlier today from Mike that it's throughout alternative pathways predominantly. This diagram relates primarily to the first and second generation drugs, bypass tracks, mechanisms of mutations within the EGFR pathway. How do we measure and discover the emergence of resistance before it's clinically obvious? This is an area of research that that's been a big part of my life the last few years, circulating tumour DNA, liquid biopsy. Liquid biopsy relates to the broader concept of any aspect of the cell or fragment of DNA that can be measured and quantitated in the blood. We think it's a better representation of what's happening to the patient as a whole, as opposed to a tissue biopsy, as you can see on the right, where the needle's put in, where you really have to be very specific. Tissue biopsies are fraught with difficulty because of where the location of the tumour is, and also you have the errors of sampling error and processing. This diagram highlights the various different mechanisms that are thought to be behind the emergence of resistance to these tyrosine kinase inhibitors, osimertinib. EGFR pathways account for about 13%. Bypass tracks, just two examples there. I've highlighted those because we have drugs targeting those, but resistance also develops to those pathways as well. On the right-hand side, you see about 50% have all the various different heterogeneous reasons for resistance. And in the middle, it's morphologic transformation that you need a biopsy for. Can you discover the emergence of resistance in the blood and then alter therapy and then delay the time to which clinical resistance can occur? Of course, the answer to that's yes, otherwise I wouldn't be for doing the Harness-1 trial. But we were blessed to see this trial in breast cancer last year. This was a landmark trial, they're called the SERENA-6 trial. And what they did in this trial is they had women who had estrogen receptor positive metastatic breast cancer on standard combination therapy. They looked for the presence of a gene that was identified as a resistance marker, ESR1 mutations, in the blood. And as they measured the discovery of that, they randomized patients who had a positive signal for this resistance marker to then be given a new treatment, a new agent, versus placebo. And on the right-hand side, you'll see this is a progression-free survival curve, extension by 7 months of the time to clinical development of progression on CT imaging. This is a case example of mine in the clinic. This is a reactive case, not a proactive case. Well, I'll explain to you what I mean by that. I had the privilege of being a part of a ctDNA study we were doing with Memorial Sloan Kettering in the US, so I was able to offer it to patients around the time of their diagnosis. So this patient had EGFR exon 21, tends not to do as well as exon 19. He was a retired GP. And at diagnosis, he had bone metastases. And he was a non-smoker. He had osimertinib, and I also gave him a drug that helps reduce bone erosion called denosumab. 8 months into his therapy, he started to get progressive bone pain, and I did a scan and found that he had progression of his bone disease. I did a circulating tumour DNA test on that, and I discovered some parallel pathways that were emerging that were mechanisms of resistance to osimertinib, so I gave him chemotherapy. And that worked for a while. The ctDNA I did subsequently showed reduction in those resistance markers until he progressed again in the liver the following year. And at that point, I actually did biopsy the liver because it was a solitary metastasis, and one of the arguments to do that is to exclude transformation into small cell and other things. And we found another mutation called NTRK, and I gave him a new drug that targets NTRK that's available in Australia. The big question for this patient in hindsight, in retrospect, was this was all reactive. We waited till he progressed, we did the ctDNA, found a reason, but we're always chasing our tail. The question is, could I have done things earlier and discovered the resistance emerging before he started to get pain, before he started to get liver metastases and the cachexia that goes with that, and his rapid decline at the end. And that's where I guess the premise of where we're going to next with the Harness 1 trial is. In the clinic today, these are the two approaches that are trying to delay resistance. They're empiric approaches. They're a one-size-fits-all. They're saying, we'll take people with EGFR lung cancer. We're going to give them a combination therapy. Amivantamab's a new antibody. Lazertinib's an EGFR TKI. We don't have those in Australia. They're trying to get that registered. That's called the Mariposa protocol. And the other one is the FLORA-2 protocol, giving chemotherapy with osimertinib. Both have shown an advantage in delaying progression and in overall survival, but they require treating all the patients with these therapies up front. The big cost to the patients is toxicity. A lot of patients have become accustomed now to be diagnosed with this cancer and have single agent therapy with an oral drug and have freedom, and they don't want to necessarily go into fairly toxic regimens up front. So the big question is, we don't have to treat all the patients like this in the beginning. How do we select who should we be treating with this combination approach? And I can tell you, I've seen patients recently who are as young as 50 with this disease, and I talked to them about this recent data and said, do you want to go on to this empiric combination? And the answer is generally not. They prefer to be on the single agent, and they ask me, can I then find a reason for why I should go on it? And we talk about ctDNA and paying for ctDNA. So to overcome drug resistance, to have a drug to be successful, we've heard about the importance of MYC earlier today. And why MYC is valuable is because it's a common pathway down the signaling pathway. It doesn't matter where you start, it's where you end up. And that's why MYC bypass signaling makes a good potential target for delaying resistance. So you need a target that is ubiquitous or can be applied across the various different heterogeneous reasons for why resistance can develop. You need to be able to attack those subclones altogether, and as I said, have a common pathway. You need to be able to treat people continuously with a treatment that's well tolerated and doesn't have limits for toxicity. We've heard previously about doxorubicin and cardiotoxicity, and there's other drugs in lung cancer we give that are neurotoxic. So we don't want to impair quality of life by doing it. We want to be able to continue it. And ultimately, it's got to be a cost-effective regimen to be validated within a phase 3 setting. So I'm going to finish up by talking about what I think is an evolutionary approach to treating EGFR mutant lung cancer. We start with upfront osimertinib. That's our standard of care. We aim to use circulating tumour DNA to identify those at risk of clinical progression. And we add or change therapy before clinical progression develops, and therefore delay symptomatic deterioration and ultimately lead to survival. And I'll show you two approaches in Australia today. One I'm a part of as a co-investigator, co-chair. It's called the Aspiration Study, and I'll briefly introduce the Harness 1 study before introducing Rod. So this study we got a $15 million MRFF grant for. It's reactive. We take patients who have an oncogene such as EGFR, but when they progress, we'll do a ctDNA, look for reasons for the progression and try to put them into a treatment silo that matches that. That, of course, is subject to being able to identify that and having the treatment. And we'll try to do it throughout the whole cancer journey. That's a reactive approach. We're not doing it before they develop a progression, but we will try to measure ctDNA in response to treatment. A proactive approach is what the Harness 1 trial is looking at. It's attempting to identify the emergence of resistance using EGFR-specific ctDNA present in the blood, and then introducing the drug that we've heard to try to halt resistance through its inhibition of MYC signaling. The endpoints of the study are primarily in the phase 1 setting, of course, to demonstrate safety, to identify the optimal dose and maximum dose of the combined regime, to identify changes in evolution of ctDNA across the patient journey, and then ultimately to get a signal for progression-free survival. We have good benchmarking from the previously published phase 3 trials as to what to expect. The study is going to be conducted in 5 sites in Australia. Will be one of them in Sydney. Monash Cancer Centre has already had ethics approved, so hopefully this will start imminently. We need to screen around 150 patients, we think, to identify the caseload that will demonstrate progression. And the initial part of this trial will look at a dose escalation phase, and then the second part of the trial will randomise patients to 2 dose levels based on the observations for the first part to try to determine the optimal dose level and combination to take forward. I was talking before, I think this could go to phase 3 at the end of this approach if we demonstrate the data that we need. So that's where I'm going to end up. Just briefly, who are we looking for in this study? Kept it simple. Really, we want patients who've got an EGFR mutation, who are stable on osimertinib for 6 months, because in that 6 to 18-month period, we know 50% are going to progress. And that's the enriched population we're targeting to try to identify progressors. They need to have evidence of positive or increasing levels of EGFR, and then the other factors are fairly standard. And I'm going to now pass on. Thank you.
Thanks very much for that, Nick. A great talk and a great presentation, I think, about the trial. So I'm not going to talk any more about the trial. I'm just going to step into the competitive landscape that we're looking at, and really just to touch on a few points that have been covered so far. It's a big market from our point of view, over $10 billion market. It's being driven by that incidence, as Nick was talking about, of cancer, particularly out of East Asia, in China there. What we know is that there's one really dominant market leader at the moment, and that's AstraZeneca with Tagrisso. They pulled in over $7.2 billion last year with that drug alone, so really significant. But there's other players coming into the market. You've got one of the pharma partners in Janssen coming in with Lazcluze. They brought in nearly $750 million last year after only having launched recently, so really making some headway there. And there's even quite a number of other companies coming hot on the heels. We think Tagrisso will still dominate, though, because they're obviously the market leader and they've got a patent expiry out to 2032. So definitely people nipping at their heels. And Nick touched on this as well. There's a lot of activity in the space now into the 4th generation. But if you also look at that incidence out of China, it's no mystery that we've got a lot of competitors also coming out of China. So a number of other companies there already on the market, approved with 3rd generation inhibitors, and already starting to push up into sizable market reach as well. So it's an active area, lots of players in the market, which is actually good for us in terms of who we can appeal to. So again, touching on what we're trying to prove out here, this is really a proof of principle for us. If we can get a result with osimertinib, with a targeted agent there, and with our preclinical data coming behind, the premise is that this will sort of prove up the fact we can combine our drug with just about any targeted agent in other indications as well. So to do that, we're really focusing on an endpoint like progression-free survival, which is something that we know regulators have approved products with before. So using something that's well known to them and understood. And again, that just means that we can appeal to the market leaders. We can improve the efficacy of their agents. We can basically help them shore up their market position. But also we can appeal to new entrants coming in as well. So they're trying to nip at the heels, so we can demonstrate better activity, improved outcomes for their patients on their drugs. They may be able to take market share off the market leader. So we can appeal to both groups. Quickly onto commercialization, which is obviously really important. We're in a highly regulated industry. We have to get through groups like FDA and EMA and so forth to get onto the market. So the goal for us is to continue with the Harness 1 study to the end of dose escalation, perform an interim analysis there, identify the maximum tolerated dose, and take the data package across to the FDA. Sit down with them, make sure the data is acceptable, that they're happy with it. We can talk about the extension of that study into an expansion or even phase 2 setting. And then also, break ground in terms of what we can do with fast-tracking the approval for the product. So just very quickly, fast-tracking is a program that came in a number of years ago by the FDA and now being replicated with other agencies. Within that, it means that if the data is compelling clinically, the agencies will allow you to bring it to the patients to get it onto the market before you've actually completed the phase 3 studies. So it's a way of, you still have to do the phase 3 studies, but you can at least be out there getting the drug into the hands of patients while you're finishing up. Obviously a huge advantage. Within that program, just quickly, with the US FDA, we know that we have sub-programs like priority review, which allows for an expedited review of the clinical program and the data by the agency. There's breakthrough designation, which allows you, as you're going through, to have more increased number of interactions with FDA reviewers about the program. It's also the program that allows you to get that accelerated approval. And now they've brought in recently real-time oncology review, which actually means that you can be giving them some of your clinical data as you're going along, and they can be looking at that rather than having to wait at the end and then have it all in one big package, which then takes longer. So there's mechanisms the governments have brought in, as I said, to try and get drug into the hands of patients a lot faster. So a great example of that is the zongertinib example, which we've got on the next slide. So zongertinib happens to be a HER2 inhibitor, basically being developed in lung cancer. So they entered the clinic back in 2021. After a couple of years, they then approached the FDA and got fast-track status approval there. Study continued on. They got the breakthrough therapy about, 10 months later, or 9 or 10 months later. Priority review followed early the next year, and then managed to get approval in 2025, both in the US and also China. So they're interacting with lots of different, a number of different agencies all the way along. So essentially, after about 4 or so years of clinical development, out of the phase 1 study with only 71 patients, a very small study, small data set there, compelling clinical outcomes though, objective response rate of 71% and progression-free survival of 12 months. So again, if the data is compelling, the regulators are quite happy to approve the drugs, noting that they've then gone on to do the phase 3 studies and continue on with development as well. So just an example there of what's possible in the current age, particularly in the oncology space. So I'll stop there. I'm trying to keep on time there if we can.
Just keep going.
Keep rolling? Okay, thanks very much.
Thank you, Roddy. Right, well, in the interest of time, we are running a little bit over, not too bad, but a little bit over. So I think we'll go to the third clinical setting, which is Dr. Anupa Kudva, who's recently joined us after a stellar career, I guess, in paediatric oncology, including working for a long period of time at Memorial Sloan Kettering Cancer Center. So Anupa's going to talk about our AML phase 3 plans. Thank you, Anupa.
To move the slides.
Yeah, just that's it.
Okay, thank you. Good afternoon, everyone. So, as Pete mentioned, I am from the US. I'm a pediatric oncologist, and I trained at Memorial Sloan Kettering. And I stayed on, and I focused on immunotherapies. For the last 6 years, I've been in oncology drug development, leading both early and late phase programs, trying to bring novel therapies back to the clinic. So I joined Racura now, I think about 2 months ago, and I will be leading the AML program, and this is what be talking about today. So I'm going to walk through our strategy of using bisantrene in this disease space. I'll talk about the landscape, the current frontline and salvage treatments, why MYC plays an important role in AML biology, the rationale for using RC220 in this space, and our development and commercialization path towards approval. So AML is a rare but aggressive blood cancer. It's defined by the rapid proliferation of immature myeloid cells in the bone marrow. Historically, it had been classified by its morphology, but now increasingly it's being defined by its molecular and genetic features. And this is reflected in the updated WHO classifications in this space. It's this molecular complexity that's driving the need for newer clinical treatment strategies in this space. And so, how do we treat patients? Unfortunately, unlike solid tumors, we're not exactly precise. We can't just go in and cut it out, resect it. So we have to really rely on chemotherapy agents. So patients are categorized as either fit or unfit, and this is based on a few different factors, usually, typically age, performance score, which is how well do they carry out their activities of daily living, other things like frailty, comorbidities, and such. So the younger patients who are more healthy are considered fit, and they're eligible for a very intense induction chemotherapy. This is usually cytarabine with an anthracycline backbone. The older patients who are older, more frail, with more comorbidities, are just not able to tolerate this, so they get lower intensity regimens, and this is with hypomethylating agents and venetoclax. And while many patients in both groups are able to achieve a remission, they're not durable, and the majority of patients do actually go on to relapse. The problem in this space is outcomes are very poor and the options are limited. So similar to upfront, patients are again classified as either fit or unfit, and the fit patients just unfortunately have to undergo very intense salvage chemotherapy. The unfit patients continue on with their lower intensity hypomethylating agents or venetoclax. If there is a targetable mutation for either for either group of patients, obviously we'll use that. And then if there's a clinical trial. And while this strategy is similar to upfront, the response rates and overall survival is much worse for these patients, just highlighting a significant unmet need for patients with relapsed refractory disease. And why really aren't there great options in this space? Well, one thing is, like I said before, we're not very precise. The majority of patients need to get chemotherapy, and this is associated with significant toxicity. I mean, this is a very heavily pre-treated patient population. Given its molecular complexity, it rapidly develops drug resistance. So all this kind of leads to durable remissions are just not common. And while targeted therapies have made improvements in this space, those mutations are only in a small subset of the overall AML population and aren't broadly applicable. And so what this really means is there's a high unmet need for agents that can be used across all the different subtypes of AML that are just better tolerated than what we have to offer currently. Had to include this because this is a cause very near to me. So where this unmet need is particularly seen is in pediatric AML. This is about 15% of childhood leukemias. With other types of pediatric leukemia, our cure rates exceed 90%, but in pediatric AML, it's below 60%. A third of the patients will relapse, and survival after relapse is pretty abysmal. It's below 40%. How it differs from adults is, like I said before, we're not very precise. We have to rely on chemotherapy, and these patients get hit very hard with upfront inductions. Unfortunately, there's also fewer targetable mutations, so those targeted agents don't really have any space to work. The other thing is, for most clinical trials, drug companies, they tend to be very cautious in including children, and so these novel agents don't get to these patients that desperately need it. Unfortunately, even when patients are treated, this comes with a significant long-term toxicity burden. So we talked about cardiac, but neurologic, psychological, endocrine, fertility complications just follow these patients if they're lucky enough to survive for their entire lifetime. And those patients who are cured by transplantation see a decrease in their life expectancy by 10 to 20 years. Now, when funding for pediatric programs is becoming more and more limited, there is just more than ever a critical need for more effective, less toxic agents for pediatrics. And so where does kind of bisantrene play a role? Well, originally it was approved in France in the late 80s for AML. It demonstrated significant anti-leukemic activity and minimal cardiotoxicity. And again, this was when we thought it was similar to anthracycline-like chemotherapy, and we know it's much more novel and specific now. In the past few decades, it's been studied in several clinical studies. Two ones that I want to talk about were in adults in relapsed refractory AML. It was used as monotherapy and in combination with fludarabine and clofarabine. And in both studies, it had an overall response rate of 40%, which is pretty good. There was also a pediatric study from the 90s, which had patients with different types of relapsed leukemias, and that had an overall response rate of 46%. There were 13 pediatric patients with AML, and 5 were able to achieve a CR. In 2018, there was a follow-up that followed 2 of the patients of these AML patients who received a transplant and are now reported as long-term survivors. They were able to carry on with their life and start families of their own, which is pretty incredible in this space. It should be noted that already bisantrene has been designated by the FDA with the orphan drug designation and a rare pediatric disease designation. But how does it compare to other second-line therapies? So this is kind of what this graph shows. The first 6 agents are targeted therapies. And the line on top, the gray bar, is basically the overall response rate that was reported in their clinical studies, and they're pretty good. The orange bar is what we call prevalence-weighted benefit. So with the targeted therapies, like I said, it depends that the patient's leukemia has to have that particular mutation, and those mutations only occur in a small percentage of the overall AML population. So when you adjust for that prevalence of that mutation within the kind of the whole population, the actual benefit is much smaller than the ORR that might be reported in the study. Now compare this to standard chemo such as cytarabine or with bisantrene. You can see because bisantrene and chemo are mutation agnostic, they don't depend on just this one mutation. Their overall response rate and the prevalence weighted benefit are basically the same. So there is broad potential across all the different subtypes of AML. The benefit that bisantrene brings is it's far less toxic than standard chemo. And why do we think it has such kind of this broad applicability? One of the things is the role that MYC plays in AML. We know that across many different cancers, MYC is responsible for cell proliferation, survival, evading kind of the immunotherapy. Specifically in AML, we know it is frequently upregulated. So there was a study, this is from a study from MD Anderson that looked at 265 patients with AML across the different molecular subtypes. And they defined overexpression as a greater than 3%. It's very small, I'm sorry. So it's this lower dotted line. But you can see over 90% of these patients had MYC overexpression. They also showed that if they had an expression of 6% or greater, that was associated with a worse overall outcome. And again, more than 80% of the patients showed expression of 6% or greater of MYC. So we know it's a central transcription driver in AML. And as it was discussed earlier, we believe this why bisantrene plays has such a strong role in AML therapy is this. When you are able to stabilize these quadruplexes in the promoter regions of the oncogenes, this leads to several key molecular effects. With the MYC oncogene, it would lead to MYC silencing. It also has inhibition of topoisomerase 2 and inhibition of telomerase. And what this does collectively is decrease malignant cell proliferation survival. It also decreases the risk of treatment resistance. The really unique opportunity that AML provides our program is a clinical validation of this mechanism of action. Like I said, MYC plays a central role in AML. We know that RC220 can help silence MYC-driven signaling in this space. Compared to a solid tumor, we're able to access leukemia blasts just with a peripheral blood draw. So we're able to correlate not only drug exposure, MYC modulation, but also clinical response in the patient in real time. And if successful, this would be one of the first clinical demonstrations of MYC-driven silencing as a therapeutic strategy. So what is our clinical strategy? Like I said, we'll be focusing in relapsed refractory AML. So we had the historic formulation of bisantrene, which is RC110. Initially, we were going to have to do a human study to show equivalence between our historic formulation and our new formulation, RC220. But based on some regulatory advice we've gotten, we're able to actually now just do a preclinical PK study instead and expedite our developmental timeline. Once this is complete, we'll be able to move on to our pivotal phase 3 study, looking at it as monotherapy. So just to clarify a little bit about our bridging study, so this will be a preclinical pharmacokinetic equivalence study. This will avoid the need for a human bridging study and expedite our timeline. And this aligns with the precedent set by the FDA in previously accepted paths. And then we'll move on, our pivotal study. So this is a phase 3, 2-part adaptive trial design. The first part, excuse me, will be dose optimization between two different dose levels of RC220. It'll be our historic MTD in AML, which is 250 milligrams per meter squared, compared to 80% of that, which will be about 200 milligrams per meter squared. And so we'll randomize patients between these two dose levels, and then we'll show our findings to the FDA and be able to establish an optimal dose in monotherapy. And this is aligning with the requirements put out by the FDA's Project Optimus. And then we'll be able to move on to our efficacy expansion, the second part of this study. And here we'll be looking at the efficacy of monotherapy RC220 versus a standard of care control arm. Exactly what that would look like still hasn't been determined. And in this space, we'll also be able to add a pediatric cohort. I would just like to say that this is actually based on a previous trial design that was already accepted by the FDA in 2025 for another drug in AML. So we're pretty confident that we'll be able to move this trial through pretty efficiently. And so, excuse me, what our strategy for AML, this is a defined orphan market with a clear regulatory path. We believe it's one of our most efficient paths to regulatory approval. It would also give us potential qualification for a pediatric priority review voucher. Now, this is an FDA program to help incentivize the development of oncology drugs in pediatrics. So if a company includes a pediatric cohort in their pivotal study and it's approved, what the FDA will do is to give this voucher, and this voucher guarantees an expedited review of a future drug they might have. The company can then keep it internal or sell it externally to pretty significant value. The last few have sold for something between $100 and $200 million, which is pretty amazing. It will also help establish our proof of concept of MYC-driven mixed signaling, the targeting of MYC signaling. We also call it MYC silencing. But what really excites us about this program in AML is not only changing how we treat relapsed refractory AML, but we think it'll change the way we treat all leukemia. And it'll open the door for expansion into other types of not only adult and pediatric leukemias, but additional MYC-driven cancers, and really make a significant difference in our patients' lives. So that's what I got. I feel like I talked really fast, but so I'm happy to take any questions.
We have found a couple of minutes, so if anyone has any questions. Oh.
Is there going to be any screening to find the 40% of patients who will respond?
Sorry. For our pivotal study is what you're asking?
So the question was, is there any way of screening to identify the 40% who would respond?
Well, in the relapse refractory setting, I don't think specifically we could, no. I mean, that's kind of why we're doing the pivotal study. And once we move on to the efficacy expansion, we'll be able to figure out a better strategy for picking the optimal patient population. And that's typically how these pivotal studies work.
Just to add to what Anupa is saying, that we're currently at building the synopsis and consulting AML experts globally. If there's any form of screening which is suggested to us, then we'll consider it, but not at this stage. It's an early planning phase.
Yeah.
So if I'm thinking about how to think about like your old data in AML and then what you have today. I guess my question is around the photoisomer, like obviously back in the 80s when you're doing the studies, like it wasn't really, you weren't aware that you had these three isomers and only one of them is working. So I guess at what point, and have you kind of gone back and thought about it, but how was the product treated in those studies? Like was it exposed to visible light? Can we assume that it was a pretty big mix of the isomers? And then I guess also thinking about the 110 versus 220, have you also, because also that would have been your prior formulation in the other studies, have you also got, I guess, head-to-head preclinical to understand the efficacy on a preclinical front, because obviously the albumin binding and things is different. So I guess helping us just trying to conceptualize your 40 to 46% is with different products, right? So help us.
I'll take that one.
Okay.
Yeah, so it's great. It's a great question. I mean, we absolutely don't know what was being administered to the patients back then. We know, as I'm coming on to my section, that the molecule does photoisomerize at about a rate of 1% per minute when exposed to ambient light. And it probably accounts for the fact that there are very different results from different centres in clinical trials. You can imagine, maybe somebody was putting on a tray near an open window and somebody else is putting it away in a fridge or something. So once it's in solution, it will isomerize. The 110 to 220 question is a great question. That formulation was actually designed to have very similar PK. So 220 gets it into solution, but thereafter it's just bisantrene going, or EE bisantrene, because that's obviously what we're interested in delivering. So the pharmacokinetics between the two. Now, I think there is possibly an argument that we could expect greater potency at a particular dose compared to the old formulations because of it converting to inactive isomers. But we're obviously looking at that. In terms of preclinically head-to-head, I don't think that has been done, has it, Daniel?
Yes, it has.
It has been done. There you go.
It's basically 110 and 220 are identical.
Yeah.
And they were designed, 220 was designed to be identical right from the beginning for exactly the reason we wanted to be able to bridge across the historical data.
Yeah. Okay, there was one further question, I think, Derek.
Sorry, identical as far as PK is concerned?
Yes.
Because my question was about the phase 3 dosing. You were going to use 110 MTD, but you didn't know the PK yet of 110. You're comparing it in a prior study to 220, but then you're taking 80% of the MTD of 110. So you have to be pretty confident that the PK is similar.
Yeah, so 110 has been used recently in trials in Israel, so two investigator-sponsored trials, and we know that was 100% EE bisantrene, so we do have that comparison. So I think we're on reasonably safe ground.
So there's really three. There's the old Ledley formulation, which is from the 80s, which is a mixture of different isomers. There's 110, which is similar to the Ledley formulation, but they're all bisantrene, and then there's 220, which is the new formulation, which is designed to solve the solubility problem. That was the original reason why it was originally dropped.
And you're confident that the prior studies of 110 MTD of 250
yes.
is correct?
Yes. Well, they may not be the correct dose. They're the effective dose. They get the same 40% response rate as the historical, even in much further advanced patients. The original studies were done in second-line patients because there wasn't really anything beyond back in the 80s, where the two studies we've done recently were in fourth-line and fifth-line patients. So much more advanced. The interesting thing from my perspective is that it's the same 40% across the board. There doesn't appear to, which is very unusual in oncology, where typically each line of therapy for probability of success falls off a cliff. So, yeah, all of this, we could have lots and lots of questions about all this, so maybe we should move
Anyway,
on.
we should
Pete
thank Anupa. Thank you. Yeah. On the side, I find myself uncomfortably sandwiched between you and drinks, but actually there's questions afterwards, so we'll have time for more questions. So I'm just going to spend a little bit of time talking about intellectual property. We did do a big webinar last September, so if you're really interested in this, you can go and have a look at that, but I've just got a few slides, because obviously IP is very, very important. Without IP, the company's worthless. So the key discovery was, as Mike mentioned earlier, the photoisomerization of the two arms, basically, of the molecule. And it was interesting looking at Lawrence Hurley's presentation that when the arms were down, they didn't bind to the G quadruplex. So this is the only molecule we believe is active. It's very difficult to prove that the other isomers are completely inactive because it's it's impossible to get a pure sample of EZ because it will equilibrate back to EE bisantrene. So it's really quite complicated. So the evidence, the circumstantial evidence is that this is the only active isomer, and it's the other isomers are not predicted to bind to G quadruplexes, and we're going to be doing more work around that because obviously that's quite a key piece of information. So the discovery of that photoisomerization, that was done in Wollongong by Mike and the team, and has led to us filing composition matter claims on the molecule. It's about delivering this molecule to patients. We want to make sure that patients are getting a uniform product. We don't want some patients getting 100% EE bisantrene and some getting 60% EE bisantrene, because obviously they're not going to have the same effect. So this is a very, very important discovery. And basically now in the modern era, you can't deliver mixtures of isomers to patients. You need to work out which one is the active one and deliver it, or prove that both have similar activity. So the importance here is that we are able to reset the patent clock, and we're building this thicket of IP around it, covering manufacturing at scale, the formulation, and obviously the EE bisantrene itself as composition of matter. And that's very, very important. It's very important for obviously the company finances, but when we come and start talking to pharma partners, this is critical importance. First of all, 20 years on a product that's going into phase 3, that's unheard of. Typically, you would have burnt maybe 8 to 10 years of your patent life, sometimes more, before you get into phase 3. So we're well ahead of the pack at this stage in terms of the patent life. The other thing is that with a formulation patent or even a manufacturing patent, somebody else might be able to circumvent your patents, develop their own formulation. Now, that doesn't really happen. That's the sort of thing that a generic company would do, but they don't do clinical studies. And if you're developing a different formulation, you'd have to go through all of the preclinical work and all of the clinical work, and you'll be trying to show equivalence or superiority or whatever with your new formulation. So that's something that you don't really see in the industry, but take it from me, Big Pharma will pick up any big stick it can to beat down the valuation on your partnering discussion. So having this, assuming it goes to grant, of course, these have been filed. We're very confident that we will get composition of matter claims, but we have to wait until we actually get those. So the longer the patent life, clearly, the more valuable, and we'll be talking about risk-adjusted discounted cash flows in just a minute. So we think we cover all three of the key areas you need to get a patent: novelty, non-obviousness, and utility. Novelty, nobody else discovered it. Non-obviousness, nobody discovered it in 50 years. And then utility, in that we've identified the active molecule. So we think we're covered in that regard. So very quickly, having skimmed through the IP, we feel we're in a strong position. We have secured, have claimed composition of matter on the molecule itself, the active molecule. We've filed multiple patent families, and the advice we're getting is that we are going to get very, very strong patent protection across this. So it really does add a lot of value to what we're doing. So that's IP. Now I just want to talk a little bit about valuation, which is a bit like sticking my head in the lion's mouth, but just a few observations, I guess. You can take it or leave it. You can buy or sell your shares tomorrow, but I thought I'd just put a few pointers out there. This is the Holy Grail on the right. This is actually the real Holy Grail, as in Indiana Jones and the Last Crusade. Everything's pucker around here. The problem with valuing a biotechnology company, and there are a few of you who do it for a living in the audience, is that the general methods using a risk-adjusted discounted cash flow is you think about the sales of the product, which are very, very difficult to predict because you're looking into the future. You don't know what the standard of care is going to look like. You don't know what the competitive landscape is going to look like. You don't know what the pricing environment is going to look like. You don't know what the geopolitics are going to do to all of that. So we're discounting a very unsafe number, and then we're putting a percentage chance of that product actually being approved, when we know that in reality, the chance of it being approved is either 0 or 100. So putting a 15% chance of making it from end of phase 2 or start of phase 3 to market is kind of meaningless. So what we tend to do in the industry, and I started out my life in investment banking, don't hold it against me, is we use comps or comparable companies or comparable deals to say, hey, well, look, this company got these terms. We think that we should get similar terms. Now, what is something that's comparable to MIC? Well, there's a lot of interest at the moment on another target. Actually, Nick Pavlakis mentioned it earlier, and that's KRAS. So KRAS sits down just under the membrane, passing the signals from growth factor receptors, so from epidermal growth factor receptor, platelet-derived growth factor receptor, vascular endothelial-derived growth factor. So all these growth factors are signaling into MYC, but they're all going through this other molecule, RAS. And when RAS gets mutated, it's stuck in the on position, sending signals into the cell. Now, KRAS is mutated in 19% of all cancers. Just remind you, take it or leave it, that 70% of cancers have elevated MYC. So both targets are very, very important oncology targets. And until relatively recently, KRAS was also thought to be undruggable. So why am I going through this? Because there are some very interesting comps out there. This is Revolution Medicines. And I'm really annoyed. I actually know one of the board members who's probably made an absolute fortune out of this. Market cap is just below 20, I think it's around $19 billion at the moment. You can see they got a lovely boost in January around the JP Morgan conference because there was a rumor that Merck or AbbVie was going to take them out for $32 billion. That hasn't happened, but it didn't do the share price any harm. They've got a number of products that are targeting pan-RAS, so they're targeting all of the different mutations in RAS. I think they've got a phase 3 readout coming up in pancreatic cancer. That's going to be very, very interesting for them. No idea what that result's going to look like. But the point is that if you are in a leadership position, targeting something that others have really struggled to target, you can create a lot of value. So that's just example number one. Example number two, Erasca, it's in the name. It obviously also benefited from the hype around Revolution Medicines. Market cap about $4.5 billion actually as of with yesterday's sell-off, and they very nippily picked up $260 million in cash in January on that run-up. So what I will point out to you is that they have 2 phase 1 drugs in development, and they've got a nearly $5 billion market capitalization. Again, just putting it out there. And then the company that really started it was Mirati. So it was considered to be undruggable, and then Mirati came out and said, hey, we've got a drug against one of the KRAS mutations. So KRAS is mutated most frequently at position 12. They managed to target the glycine to cytosine mutation at position 12, so G12C. G12C is about 15% of all KRAS mutations. It happens to be the majority of lung cancer mutation. So when it's mutated in lung, more often than not, you're going to get G12C. Now, what I'll point out are 2 things. One is the valuation, that 2 years after starting their phase 1 trial, they hit $12 billion market cap because they were considered to be in a market-leading position. And then also that 4 years later, they got approval. And I think from memory, they got approval on 112 patients' worth of data. And this is because there were no drugs out there, and this was an identifiable mutation, and they had a drug. They lost a lot of value because Amgen were very, very quick in, first of all, chasing them, and then overtook them. They actually had a few regulatory issues that delayed their filing with Mirati. So again, if you've got something interesting that is clearly working, where nobody else is there, you can get approval very, very quickly, and you can generate a lot of value. Obviously, we need to get this message out. We're just at the start of that process. And just picking up on what Rodney said earlier on about zongertinib, it wasn't alone in the second half of last year. There were 3 drugs in non-small cell lung cancer, 2 of them targeting a tyrosine kinase linked to HER2, and one of them was a EGFR tyrosine kinase inhibitor. And I just point out that these were approved on early stage trials with relatively small patient numbers. Now, I'm not telling you that we're going to be able to follow that path exactly, but I think these act as precedents for us going to the FDA with our data from early trials and saying, right, what do we need to do to get approval? And they may not be really, really big trials. It might be that we show a good response rate in patients who basically are not going to have any other targeted therapy, then we may be able to get much faster approval. Again, we can't promise that. We haven't had those conversations with the regulators, but I think these observations are valid. So this is just a list of MYC inhibitors in development. There aren't that many of them. So there are now well over 100 KRAS drugs in development. There are loads coming out of China, as well as from other biotechs around the world. We've put ourselves very kindly at the top there of ones that are having a direct impact on MYC. It's not a direct inhibitor. That would be a small molecule interacting with the protein, but either silencing the gene, causing the protein to be destroyed, various methods that are being chosen. No one's really made much progress yet, but do bear in mind that we have all that historic data, so we actually do know that our MYC silencing drug does work, and we do know that it's safe. There are other pathways, but the thing to observe here is there isn't a single big pharma company on this list. Not one. So they've been trying to do this for many, many years. As many of you know, I was on the board of a company called Micarex targeting MYC for 7 years. I stepped off that board because of the clear conflict of interest with Racura. But this is a very, very interesting area for us to be playing in. But the competition is fairly limited. And the two truly direct inhibitors, the peptides, they're peptides, which means they're not very good drugs. They're not very drug-like. And the results have been modest, stable disease as best response in those trials, almost certainly to do with the lack of drug-likeness of using a peptide. So I'll just finish now with a corporate snapshot, because at the end of the day, all this wonderful science, all these lovely programs, we've got to pay for it. So we finished the half year with 20.9 million in cash, and we still have roughly around 17 million. At that time, we had around 17 million of piggyback options still to be exercised. They exercised at $1.25, expired 26th of May, so not too far away. So hoping that they will be converted. With the cash we have in the bank at the moment, we will be able to complete the Harness 1 trial and the CPACS trial. In terms of the AML, to really get moving with that, we need more of that option money to come in. We won't start any program until we've got enough cash to finish it. That is stupidity. That's like committing suicide almost. There's no point in getting through 3/4 of a trial and then running out of cash and not being able to complete it. You might as well have never started it. So we definitely have the discipline at Racura of only starting programs when we have the resources. Hopefully that'll be before we get to the end of May. So with that, I'll leave you just those little thoughts. We can open up a broader Q&A session. So I'd like to go to all the speakers to come up and sit at the front. Right, so again, thank you very, very much indeed for attendance and and for being here. Any questions?
I'm very much a non-clinician. I try to read most of the announcements and pick up knowledge. I have a finance background, so the question relates to some of the earlier slides where there were the results from the tests on mice, and specifically combining RC220 with other drugs. What wasn't clear to me was what happened after the drugs were stopped or continued. They seem the trials on mice seem very short term, and it wasn't clear to me if under any combination the cancer had been cured, because it seemed that the poor mice eventually passed away. So is there a cure, or is it just a deferring of the symptoms?
Who wants to take that one?
Maybe I should have gone to Wikipedia. But
yeah, let me just be upfront about that. I think the objective, particularly we're looking at Harness 1 in lung cancer, which I think you're referring to that combination with osimertinib. I think the objective there is to delay treatment failure. I think the idea that we're going to be curing large numbers of patients in that trial, that's kind of not what cancer does at that stage. So I think we have to be realistic, but there is still value in extending that. I don't know, Nick, do you want to respond?
There's limitations in the animal models. I spoke with my PhD and I'm doing some great experiments, and that's similar. You delay progression in a lot of the times. You can find models where you can look and see if you can get cures, but that doesn't always translate in the clinic. But there's a value in delaying symptomatic clinical progression. When you have a patient, that first part of their journey is the best journey. Once they start to progress later on, their quality of life is impacted greater. So if you can extend that first part of the journey from the remission they first obtained as long as possible. That's still a very valuable outcome.
So for humans, 100% cures are not off the table.
No, in oncogene-driven cancer, and I treat all of them, we'll see patients who are alive at 10 years. What's a cure? A cure is you outlive the disease and you don't die of it. So I've seen patients 8 to 10 years with EGFR cancer. Most, though, will live several years. And so we're trying to extend the majority in that median. Right? There will be some patients that we call them exceptional survivors. Only immunotherapy in the metastatic setting has what we've created, this tail of the curve, where people we think might have outlived the disease, but we haven't even had long-term follow-up on them yet. So I've got people at 8 to 10 years who are still wandering on the edge of the seat will have relapsed. We don't know yet. When they die of something else, that's when we call it a cure.
Utterly macabre, but... and the other thing just to point out there is that, you know, we've been pursuing this angle for a relatively short period of time. So some of these experiments are, you know, look-sees, you know, do we see an effect? And, you know, you'll see more data coming out from the team, you know, with different combinations as we explore this. But from a business development point of view, I want to know, right, do we think it's realistic that this could work in combination with other drug types, not just the EGFR drugs, because that then expands the number of companies we would talk to. So yeah, so you know, we're at the beginning of this process, and you know, science is, you know, imprecise. You have to go and do some pilots, and then you see something, and then you explore that further.
So I had a question about the Harness 1 study, specifically the type of patients you're recruiting. And help me if I misunderstood. Are these patients, are they first-line patients who are still on Tagrisso, when you're recruiting them, then you're looking for a signal of progression, and I guess firstly what that signal of progression is. So then are they technically second-line patients as you're bringing them in, or just help me place them?
No, they're first-line non-progressors. They have to be non-progressive on Tagrisso to start with, and they have to demonstrate presence of ctDNA EGFR. We know ctDNA clearance is a good prognostic factor. Because we won't have captured the patients at the beginning of the journey, you're capturing at a different time point. You're looking for magnitude of ctDNA and also elevation on serial measures. So if you demonstrate that, you know they're heading up towards a progression event, and that's when you're trying to capture them.
And how long are you measuring that, I guess, that elevation over from when you first see their baseline ctDNA when they come in?
You've got to be at least 6 months on Tagrisso, and then if you're disease-free, you can capture patients who are 12 months down the track. Because one of the challenges is there's a bunch of people out there who are in remission currently on Tagrisso, and trying to capture them and enrich the study as much as possible with them is the goal, because you fast-track the study if you can get them on. So if you narrow down the selection process too much, it'll take you too long to accrue. So if you pick 6 months, as I said, 50% will relapse by about 18 months to 2 years. So we're going to give enough of a window to try to see people in that. Because we don't know what the gap is clinically, as in the breast study, which was 7 months between clinical progression. So I think there's enough leeway there to pick them.
Yeah, I guess to add to that, we're going to be monitoring patients serially, so longitudinally. So when they come into the first part of the study, we're looking at doing roughly every 6 weeks. So we'll be tracking them as they go along to try and capture the time where they start to increase, hopefully prior to an imaging progression time point.
Okay, yeah, so I guess I'm trying to...
Before that happens, before they actually technically go second line.
Okay, so then if we're thinking about some of the bispecifics that are kind of coming in, and there's the ivonescimab and those ones people are excited about, these patients you're capturing are before they would be eligible for those studies in a TKI failure setting?
Yeah.
Okay.
Yeah, exactly.
Yeah, and the idea is that, and part of the attraction of getting people onto it, we extend the utility of osimertinib, hopefully, and then those patients still have all of the treatment options they would have had they not gone on to the RC220. So we've just given them, hopefully, I don't know how many months we're going to get, but we'll give them a period of very, very high quality life.
Yeah, sorry.
It's also about avoiding unnecessarily treating everyone up front with combination therapy. The big advantage to the patient is they see this as a rescue approach as well. Not everyone wants to. The 2 approaches I showed you up front, they're empiric. give them amivantamab, and they're toxic regimens, and give them chemotherapy, and people aren't liking that. So this is offering an alternative approach. Start with osimertinib. Then if you demonstrate this risk of progression on ctDNA, we'll salvage you, hopefully, by giving you this treatment before clinical progression development.
I had one other.
Oh, sorry, I was just going to add, we know from a publication of Gray et al. From a number of years ago that looked at the osimertinib studies, we get about a 3 months head up from the point in time where ctDNA increases versus when imaging progression occurs. So that's that sort of window we're trying to slip into there.
And I just had one other question. This is a bit broader about MYC. So you're talking about 70% of cancers kind of have this upregulated MYC, but upregulation is not necessarily like, sometimes it's a driver, sometimes it's just a downstream effector. And things like KRAS and WNT and all of those that you're speaking to, they also upregulate MYC, but they aren't the driver, right? So targeting MYC won't necessarily help in those cancers.
Well, no, I think if you do look at the patients longitudinally, and we're looking at some data, real-world data, you see that MYC is high prior to treatment in the tumour. We don't measure it if the tumour is responding, because it's very, very difficult to biopsy. So we go into a bit of a dark patch. But what we do know is that on relapse, the MYC is as high or higher than it was before treatment. So something else is picked up. And you're absolutely right. Wnt, beta-catenin, JAK-STAT, all these pathways impinge on MYC. So it might not be a driver. So KRAS is a driver, but it's a very, very, very core component. There was actually a paper out fairly recently that said, I was looking at tumours with mutant RAS, I think it was G12C, and they showed if you knock MYC out or you knock the mutant RAS out, it had exactly the same effect. They phenocopied one another. So it's almost like there's a direct pipe between RAS and MYC, and if you stop MYC, then you're going to have the same effect as inhibiting RAS. I think the art is to try and strangle MYC as hard as we can for as long as we can to choke off all of those pathways, all of those certainly canonical pathways that are involved in cell signaling. So yes, you're right, it's not a driver, and we do think that the fact that we're almost certainly binding to other G quadruplexes, and G quadruplexes tend to be regulating important genes, as in genes that if they're uncontrolled are going to cause a problem, like MYC. So it may well be that we're getting a broader effect by targeting different G quads. That's part of the research program we're embarking on at the moment. So it may not just be MYC, there may be other components. But again, getting back to what Mike said with the looking at the gene pathways, clearly the heavy lifting is being done by silencing MYC. That's the signature we're seeing across all of the data. So there may be other components, but we do think the major effect is through silencing MYC.
I would just add that MYC is really an accelerator. So it's not really a driver. It doesn't actually make a cancer cell, but if it's present, it makes the cancer cell far more aggressive and faster growing. And the other one too is it sits at the bottom of so many different pathways, and that is what we've seen empirically initially before we knew all about the G quadruplex. We saw that it worked with all these different kinase inhibitors that hit all these pathways that all converge on MYC, and we saw this synergy. So we're not, apart from AML, we're not planning, in most cases we're planning to use it in combination to really help maintain this focus on trying to prevent and delay resistance from arising to these really effective drugs is, I guess, it's an add-on to what already exists and making those things that already exist work better.
On the diagram of different cancers with overexpressed MYC, the highest one was pancreatic at 97%, but race isn't doing anything in pancreatic cancer. Please comment.
Pancreatic cancer is where good drugs go to die, unfortunately. It is so difficult to treat, and the reason why is not that you can't treat pancreatic cancer, is that you can't get your drug to the cancer, and because it gets surrounded by fibrotic tissue, which just basically excludes everything out of it. It's very, very aggressive. It grows incredibly quickly because MYC's massively upregulated, but you just can't get any drugs in there. So you saw Amplia came out yesterday, was it, or the day before? And they were talking about getting 5 CRs out of 64 patients in combination with the most aggressive chemotherapeutic regime you've ever seen, and seeing this as a great win. And that's because you just can't get anything into these cancers. So whilst pancreatic cancer will probably, someone will eventually go and look at, see how does RC220 work in pancreatic cancer. Hopefully it's not us.
Yeah, I'll just actually add to that, and I know Chris Burns very well. I sent him congratulations yesterday on the share price doubling. Well deserved for him. Biologically, if you look in the literature, combining something that inhibits MYC with a FAK inhibitor would potentially be very potent. So I keep saying, come on, let's try it together. So yeah, at some point, we're just like KRAS has fallen, hopefully MYC is now about to fall, we will find something that works really well. I remember back in 95, no, it wasn't 95, sorry, in 2000s, going to ASCO when the first ipilimumab, first checkpoint inhibitor data came through in melanoma. There'd been no progress in melanoma for decades, literally decades. Stage 3, stage 4 was a life sentence, and then all of a sudden it transformed into one of the most treatable cancers because it had lots of mutations that the immune system could finally see. So, we have these dogmas, you don't do pancreatic, but then at some point you crack it. And, that's always the case in medicine. There are some, very, very difficult things to do, but once you manage to do them, you change a lot of patients' lives.
This question is to Nick on the Harness 1 trial. So from what I could gather, this was more to do with phase 1 line 1 patients. That's where the RC220 regimen would fit in. Is that normal, that where you could put somebody on a trial right after their first line instead of putting them on line 2 or line 3 of treatment?
Yeah, you can in this case because you're trying to extend the progression-free period from the first-line therapy. And so you're having an intervention and you're trying to select the patients you think are at risk of doing that. So it's both ethical and it would be acceptable by patients and certainly gone through ethics already for that purpose. And in my thoracic oncology group, we have quite a few lived experience patients with EGFR, and they're all seeking that exact same thing. How do we extend that progression-free period on our first-line therapy rather than wait to fade out? Not everyone has a simple journey. Some people can present when they progress with a single tumor. We can treat them at this time therapy. But once you get rapid progression, it could be MYC-driven, and you get this bypass pathways across all the tumors in the patient that can progress quickly. I mean, I have to get second-line or patients. It's not a given that you'll get second-line therapy in the real world.
Chris.
Just got a quick question for Dr. Messina. In the CPACS trial, I'm just trying to understand the logic of using salvage therapy patients in the phase 1A to determine the maximum tolerance dose, and then going into anthracycline-naive patients in 1B. That's one part of the question. And the second part of the question is, given that it's a tumour-agnostic trial in the solid tumours, what does an efficacy signal look like? Or rather, how do you judge the strength of an efficacy signal to then determine what you're going to be targeting?
You go. But this way, yeah. So taking the first part, it's because it's a phase 1. Yes, the patients we've got multiple different tumors. That's absolutely true. They have salvage therapy, maybe for some of them, but most of them it's more quality of life perhaps or something of that nature. But we're there to just determine the safety, and they get access to a drug that may work. So 1 N equals 1, and it doesn't have to be that's not an efficacy-based study, essentially. We just look at, the reason I mentioned efficacy is that we just start to get some of the signals of efficacy. We'll check some of the patients, see what their tumours are doing, but as a whole, they're mixed population. The second population is also mixed at this point in time. I did mention that in there's an interim analysis where we'll really probably look at the data and see if we can refine that population that are anthracycline naive. So there's a potential there. So that's your second question, exactly.
Given that there's going to be a mix of solid tumours in that second phase.
Okay, yeah. So in that second phase, at the moment, there's a mix, but that might be the case once we do the interim analysis. We might stratify by indication. We have to work out that part of it. But then at the moment, it's planned. The reason it's the way it is at the moment is because we had efficacy approval, which is an advantage when we're doing one study with two different population groups. That's not normally the way it would work. They'd like to understand exactly what your question is, what doing it, but we got ethical approval, so it gave us an advantage with the proviso that we would do this major interim analysis, make a proper assessment, and then reapply to the ethics, the actual refinements.
So theoretically, you could pursue a couple of different types of tumours. Yeah, great. Yeah. Thank you.
Good. Right. I think it's my Tanya, last one. Okay. Yeah.
So just one on the choice of, like, all of the work you've done is with the AstraZeneca's drug Tagrisso. Is there a reason to think the results or the efficacy will be different when you combine it with a fourth generation drug or a third generation drug? Can you comment, please?
I thought the two compounds, the Chinese compounds, were very similar, the third generation drugs. I haven't seen anything that indicates superiority of those. They weren't designed that way. They were really designed just to be competitive. So biologically, some of the fourth generation drugs are designed to be, as they have, I guess, a longer efficacy of action. But up until now, we haven't really seen that yet. That may change. The drug, the combinations that have shown superiority at the moment have actually been lazertinib and amivantamab. So that's lazertinib, an alternative drug, but it's more toxic than osimertinib. One of the reasons why we're sticking to osimertinib is actually it's a really good backbone, very well tolerated. And then the other combination I showed you in the two trials, which is an empiric approach, is chemo and osimertinib, but all the toxicity is largely added by the chemo, and it's continued in a maintenance fashion as well. So where Aussie stands out at the moment is in medicine, medical doctors tend to be a bit conservative. We get used to a compound, we stay with it. In Australia, we only have osimertinib, so that's our only third generation drug. Or those of us that used lazertinib so far don't think it's that great from a tolerance point of view. There are some that look comparable in the Chinese market, but we're not going to see them. I don't think necessarily, if we prove principle in this compound and you get a drug very similar, theoretically it should be very similar, but that'll have to be shown in a subsequent trial.
Right. So I'm calling the end of the meeting here. It's my job as chairman. I failed to keep it on time, but I think I'm now eating into valuable drinking time. So we will have drinks outside, and you're free to, as I said, quiz the team. Just want to thank you all for being here, giving up an afternoon to drown in science. Isn't everyone's idea of fun? So I appreciate you being here. Thanks to the team. A lot of people have contributed to the slides and putting it all together. And I also want to thank IR department, so Kitty, Cherie and Jane, and also Sophia. Sophia, VP of Ops and Strategy, has spent an extraordinary amount of time putting this all together and refining the slides. So thank you to you, Sophia. And hopefully we've left you with an impression that this Racura is a company that's really going somewhere. We've got some very interesting opportunities ahead of us as we work through on this mechanism. As I said, it's still very early days. It all feels new. And hopefully pleasant surprises will come thick and fast as they have over the past 12 to 18 months. So with that, I'll close the meeting. Thank you very much indeed. And I'm sorry, I failed to do it. Thank you very much to Nick and Erin, our guest speakers, KOLs, advisors, for your contribution as well. It's been brilliant. Thank you.